Hello everyone, so let me know if your side audio video is clear, because on my side it's clear now. Hello everyone, so let me know if today we are going to learn about the muscle physiology and basically what are the proteins and how they are going to keep our muscle intact and on their side. Yes, thank you, MI. So let's start with the muscle physiology. Rana Prem Shankar, hello. New names, welcome bacho. I was wishing that today there should be more students because otherwise the weekend is going to get ruined. But thank you all for joining, right? So we are going to enjoy today because it's a weekend and I promise you it is not going to be a boring class, right? So let's start with the muscle physiology. First of all, I would like to start the class with the wonderful message that I can't do everything, but that I won't stop me from doing the little I can do, right? So whatever little we can do, we have to do. Hello Ankur, very good evening. Happy weekend to all of you, right? So let's learn little by little whatever we can in our control, whatever it is there, we are going to do it. Okay, let's start with some recap. Yesterday I have told you that there is going to be, actually it was a GIF, but it is not going to work now. Yesterday in the GIF we saw that this is the nerve, and through the nerve what is going to happen, the action potential will be carry forwarded. As soon as this action potential is carry forwarded, what is going to happen? There are going to be voltage gated calcium channel. Hello Jai Kishan, very good evening. So this action potential is going to carry forward towards the axon terminal, and we know there are going to be one type of channels. What are those channels? Those are nothing but the calcium channels. How they get to know that the action potential is there? Because they sense the voltage, right? As soon as there is a voltage change, there is going to be the activation or opening of these calcium channel because they are voltage gated, right? So their gates are going to get open as soon as the voltage is going to reach to them, right? As soon as the Shah Rukh Khan and he take a descent from the helicopter, his mom is ready with the thali, right? Like that, the action potential, as soon as it is going to come towards the axon terminal, the calcium is ready to open. And as soon as this is going to open, we know that outside of this nerve there are more calcium ion as compared to the exoplasmic calcium. So that's why, because of the gradient, as soon as these doors are open, this calcium, they were waiting for the calcium channel to open and they're going to enter into the nerve. As soon as they enter, I told you yesterday itself that yes, I'm Marathi, and how this comes in between the action potential. We don't want such action at this platform, right? So what is going to happen? As soon as it is going to open up, definitely the calcium is going to enter, and the calcium, it has two major roles in the nerve muscle physiology. One is in the nerve, one is in the muscle. What is the function in the nerve? Exactly. This is nothing but the exocytosis, right? It makes the vesicles to go out. As our own faculty, senior faculty, they ask us to come out of the room and take the class on the weekend. So same, it's a job of calcium. It's like they are like parents. So what they are going to do? They are going to take a stick and they are going to make a study, right? So same is going to happen with the calcium. The calcium is going to enter in the nerve and they release the neurotransmitter via causing the exocytosis of the vesicles. As soon as the exocytosis of the vesicles, that is going to happen, of the neurotransmitter. Which neurotransmitter that we are going to talk over here? We are talking about neuromuscular junction. Answer the bacho. Which neurotransmitter do you think we are talking over here? So what are the neurotransmitter? Which is the neurotransmitter in the case of this neuromuscular junction? This is the question for you guys. Yes, I want the answers from you guys, so it is going to be quicker. And then what is going to happen as soon as that neurotransmitter that you're going to answer? Yes, MI, absolutely wonderful. So I got the answer over here. Chocolate from my side to you, right? So that is okay. that is going to sit on this channels over. Now is it clear, bacho? Just 2 minutes. Just 2 minutes. Yeah. Now it's clear. Is it clear? Yeah. So let's start with the next thing, that Arif Ali, Atul, now is it there? Voice, I guess the voice is back now, as you guys are back on the comment box. Now, okay. So let's start that the sodium is going to enter, and where this is happening at the junction. And this sodium channel, remember, they are present on the membrane of the skeletal muscle. We call it as sarcolemma. The sarcolemma is going to get depolarized because of this opening of sodium channel, and as soon as it is going to depolarize, they are going to carry forward that depolarization as soon as it hits the threshold potential, isn't it? So it is going to produce the action potential, and that action potential will be carry forwarded. As you guys are answering, there are more and more student entry I saw, right? And the more and more messages I'm getting on my personal window, thanks to you guys, right? So action potential, as you start to study, your surrounding people, they start to study also, if they are studious, right? Otherwise you will leave the studying. So that is going to happen. So choose your friends wisely. Why? Because your surroundings matter, right? You are the average person of your surrounding. Okay, now we talked about sarcolemma. Whatever we talked just now, that is on this part, right? So this is the sarcolemma that we were talking about. Okay, there are going to be sodium channel. Now what happens as soon as the action potential is produced, it is going to go in the tubule, and that looks like a T tubule, isn't it? This looks like a T. So please remember that this is going to go in the T tubule. That action potential will be trickling down into the T tubule, and we know there is going to be triad in the case of skeletal muscle. What is the triad? Triad, the name itself suggesting that there must be 3 things. So what are the 3 things? I'm making 2 arrow. That means one will be T tubule. There are going to be T tubules, and what you can see over here on the side of the T tubule, in the pink line I have mentioned it, or I have highlighted it, that these are 2 L tubules, or they are also known as terminal cisterns. Okay, so they are the cisterns that are present on both the sides of the T tubule. Please remember that is going to be only and only present in the on both sides in the skeletal muscle. So there are going to be 2 L tubules, and that makes the triad, isn't it? So skeletal muscle, triad. My question, where you can see the dyad, that means 1 T tubule and 1 L tubule, right? So this is seen in the Hindu marriages, only one guy can marry to 1 woman, right? So where this is going to be present. Okay, Rana Prem Shankar, hello bacho. Arif Ali, Atul Babu, right? So let's move on. That is my question to you, that dyad is seen in. Recently, in one of the university exam, this was asked that the dyad of this system, sarcotubular system, is seen in which of the following muscle? Obviously, your questions are going to have options. I'll give you the option: skeletal muscle, smooth muscle, cardiac muscle, all of them, right? You already have 50-50 because we just now discussed the skeletal muscle. So skeletal muscle is out. The all of the above is also out, right? Yes, Kamlesh, you are absolutely excellent. Audible now? Is it audible? Forward करके देख लो एक बारी. Kamlesh, is it audible now? Let's move on. Okay, so in the cardiac muscle, I was telling you that one T tubule and one L tubule is going to be there. Okay, so thank you, Kamlesh. So let's move on. So the tech was teaching us the patients, right? Now, I already told you over here also this GIF is not working, but I'll tell you later that as soon as this is going to, the action potential is created on the sarcotubular system, what is going to happen? Definitely it is going to trickle down into the T tubules. And we know on the T tubules which channels are going to be there. In the last class we have discussed, just tell me which channels are going to be there on the T tubule. So if I draw the T tubules like this, and on the one side if I draw the L tubule like this, and L tubule terminal system, this is the terminal part of the sarcoplasmic reticulum, isn't it? So sarcoplasmic reticulum, so in the, let's take a black color over here. So there are going to be some channels. I want you to answer what are these receptors called as on the L tubule. So definitely the action potential will be produced here and it will be trickled down, right? The depolarization will be producing action potential which is going to trickle down and stimulate one receptor. What is that receptor? Yes, answer. Prakash, Shravan, thank you. Rana, chalo answer do. What is the channel name? So A, channel A, I'll ask you what is that channel. I want the answer from you guys because yesterday in the class it has been done, right? And there is another channel, right? Channel number or let's say channel B, which is going to be there. Very good, Kamlesh. Very nice. So yes, you are absolutely correct. On the T tubule we do have DHPR channel, that is nothing but dihydropyridine receptors. Okay, and they are connected mechanically. Please mention it. It is connected mechanically on the gate of this second channel, which is known as RYR channel, ryanodine receptor, which is present on the sarcoplasmic reticulum. Wonderful, Kamlesh. Wonderful. What is going to happen? Remember, this is the receptor which is not going to get open. This is not the channel in the case of skeletal muscle. Why I am stressing too much? Because when we are going to read about the difference between the skeletal muscle and cardiac muscle, definitely it is going to be different, and that's why I'm stressing upon these things. So this is going to be attached with the spring-like structure with the RYR. That means they are having mechanical connection or physical connection, right? Yeah, mechanical connection or physical connection that they're having, bonding is there. Okay, now in the cardiac muscle also they do have bonding, but this is not direct bonding, it's indirect bonding. Okay, just as we have with other friends and all, and the special friend that we have, that is the difference between the cardiac muscle and the skeletal muscle, right? So these are the special friend, that is special wala friend if I ask, right? So this is the best friend. So they do have the physical connection in between that is going to open up. As soon as the DHPR is going to get activated, you can see over here that I'll just tell you. This is the nerve ending will release the acetylcholine, which is going to activate these cationic channel, you can see over here, but the sodium gradient is much more, so that's why we call it as ligand gated sodium channel. As soon as the action potential, that means it is going to produce the depolarization followed by the action potential if it is reaching to threshold. This action potential will trickle down towards the receptor that is known as DHPR channel. As soon as the DHPR is going to get activated, I'm not saying open, it is activated, the spring will coil, and because of the coiling of the spring, you can see this was the resting phase over here, and as soon as I stimulated it, what happened over here, that the spring will coil and that will make the RYR channel open. And we all know that the sarcoplasmic reticulum is a bag of calcium. So definitely what it is going to do, it is going to release all the calcium. Calcium is waiting to get out, right? So it is going to come into the sarcoplasm. As in the cell we do have cytoplasm, in the axon we have axoplasm. Similarly in the sarcoplasm, that means in the sarcotubular system, once it is going to get stimulated, it is going to release the calcium into the skeletal muscle ka plasma plasma content, that is nothing but the sarcoplasm. As soon as this is going to happen, this cross bridge cycling will be activated. What is it? We are going to go in the detail. Remember, from the muscle we have come down to sarcotubular system, and in the muscle we are going. As soon as we move into the lecture forward, we are going to zoom it in each of the proteins also and their connections. Okay, so we are going inside the muscle fibers again, right? So Ritesh, I got your answer. You are also right. So please remember that this is the mechanical connection. That's the only thing you have to remember when it comes on to the difference. Now what is the difference between the skeletal muscle and cardiac muscle? That we are going to see over here. Now remember, if I talk about the cardiac muscle, let's suppose this is not the skeletal muscle, there will be no spring in between. That means there is no physical connection, right? As professionally we do have, we have to talk with each other, right? So what they are going to do, that these DHPR channels, now I'm saying channels, they are not only receptor now, they will be channels in the case of the cardiac muscle. And as soon as the action potential will trickle down towards the DHPR, if their channel, what is going to happen? They are going to open up, right? So as soon as they open up, the calcium entry will happen in the case of cardiac muscle, right? And as soon as that is going to take place, this increase in the sarcoplasmic calcium, it is not going to be that high as high it is there from the release of the sarcoplasmic reticulum, but definitely around 20% calcium is going to come from the ECF, right? Because outside, just outside of this T-tubular system, it is going to be the ECF which is going to be present. That is nothing but the interstitial fluid, which is the part of ECF, extracellular fluid. Now, this increase in the, a little bit increase in the calcium level, they will be sensed by the RYR channel. Okay, and they are going to open up automatically in the case of cardiac muscle, isn't it? So I'm going to rub it off, because again you will be confused when I'm going to give you the PDF. So let's draw that with our own beautiful hands, right? So yes, as soon as the action potential will be generated, what is going to happen? Let's say these are the sodium channels, mainly cationic channel, that is going to produce the action potential, right? And that action potential will be opening up the channels which are nothing but the DHPR channel, and this DHPR channel will be causing the entry of calcium. But how much calcium? Remember, it is not much, it is only 20% of the calcium which is going to come from the ECF, that is extracellular fluid, right? As soon as this is going to come, there is going to be the L tubule which is going to be present over here, and on that also we do have channel, that is nothing but the RYR channel again, right? And remember that this RYR channel is going to sense this increase in the calcium level. As soon as it is going to sense that calcium, it will also open up, and that will cause the calcium release. But this release is going to be around 80%. That means if I ask you the maximum calcium in the sarcoplasm comes from, either it is skeletal muscle or it is cardiac muscle, your answer is going to be sarcoplasmic reticulum or RYR channel itself, right? The only difference over here is that there is no mechanical connection, just it is happening because of the increment in the calcium. 20% is going to come from the ECF, but remember, this is going to be very essential part. If this calcium, this 20% is very less amount, but if this lesser amount is also not present in this sarcoplasm, what is going to happen? There will be no opening of RYR, so rest of the 80% will not be able to come into the sarcoplasm. So that's why the smallest steps are important. Just to begin, it is important to initiate, right? As soon as the momentum is going to form, you'll automatically studying on your own. So same is going to happen in the heart also, that is the cardiac muscle. This 20% lesser amount but essential to open up the RYR channel, and again as soon as the calcium is there, the contraction will start. Okay, now if I talk about the muscle protein, it is going to be similar in the case of the skeletal muscle also and cardiac muscle, but there are differences between skeletal muscle and smooth muscle that we'll learn later on. Step by steps, let's write it up, what is going to happen. So quickly, action potential will be there. As soon as the neurotransmitters are released, this action potential will opening up this action potential, let's say in the nerve, causes the exocytosis of neurotransmitter. As soon as that is going to happen, with the help of calcium channels on the axon terminal, isn't it? So calcium, please remember at least the two roles in the nerve, that is exocytosis of the neurotransmitter. In the muscle, it is going to be causing contraction. That is going to stimulate or open up the ligand gated. As soon as this neurotransmitter sit on the receptors that are present on the muscle, they are going to open up the ligand gated cationic channels, mainly cationic channels, mainly the sodium channels. Sodium going to trickle down, so that's why in the most of the books it is written as sodium channel. Now the sodium is going to create depolarization followed by action potential as soon as it hits the threshold, right? So action potential will be formed, and this action potential will be trickling down in the T tubules. In the T tubule, it is going to open up. Now I'm writing about the cardiac muscle, so I'm not going to just write the stimulation, but I'm going to write the opening of this DHPR channel. So opening of DHPR will cause the entry of calcium, right? So this will lead to entry of calcium from the ECF into sarcoplasm. Though it is the lesser amount, but essential. I hope you can see that. But essential. Why I told you lesser amount? How much lesser? Only if I take calcium, which is required for contraction, is as 100%, then it is going to be 20%, but it is essential because without this the RYR won't open up, right? So as soon as the DHPR is open, calcium is entered into the sarcoplasm, the RYR will sense the increment or increase in the calcium in the sarcoplasm, which came via the ECF, right? And as soon as that is going to sense, these RYR are going to open up. Opening will take place, and we know these are present on the sarcoplasmic reticulum. It is a bag of calcium. It is going to release the calcium. So calcium is going to get come into or comes from sarcoplasmic calcium entry. Let's write calcium entry into sarcoplasm from the sarcoplasmic reticulum. Okay, so in the sarcoplasmic reticulum is present. So from the sarcoplasmic reticulum, we are bringing the calcium into sarcoplasm. As soon as that is going to come, there will be removal of papaji. Who is the papaji of myosin? Tropomyosin, which were not allowing the actin and myosin to meet, isn't it? So tropomyosin will be removed via this calcium will be binding to troponin, right? And this tropomyosin will be removed, or it will be displaced. We can say displacement of, better word will be displacement. So we can write over here removal rather than that. We can write displacement of tropomyosin, and actin myosin cross bridge will be formed. Finally, we distract the papaji and let the actin myosin meet together, right? So cross bridge is going to form over here. Okay, so actin and myosin cross bridge is going to form over here. Let's move on to the next thing. All right, let's move on to the next thing, that is the EC coupling. We have learned about the EC coupling. First thing, there are two differences, major differences. What are the two differences between skeletal muscle and cardiac muscle? So please remember, in the skeletal muscle and cardiac muscle. In the skeletal muscle we have triad, on the other hand in this we are having dyad. Okay, that means only one L tubule will be present in the case of cardiac muscle. Then there is mechanical coupling. Because of which the RYR are going to open. That means the source of the calcium, I'll just write in between so that it will be easier. Source of calcium. Over here in the skeletal muscle it is going to be sarcoplasmic reticulum only. On the other hand, it is going to be sarcoplasmic reticulum mainly, that is 80%, but for that we require ECF calcium, which is going to be essential, which is going to cause the release of the calcium from the sarcoplasmic reticulum, right? There is no mechanical coupling which is going to be present in the cardiac muscle. Now there are types of this RYR channels also. How to remember? I have one mnemonic for you guys, that is SM followed by SM. What I mean by that, there are three types of RYR present. RYR type 1, type 2, type 3, right? So what is type 1? RYR type 1, type 2, type 3. S, but it is not followed by M, so it is going to be not smooth muscle but the skeletal muscle. So it is going to be skeletal muscle, right? Then M for myocardium. We can write cardiac muscle, but then it won't fit into the mnemonic, right? So then it is going to be called as myocardium, right? And the smooth muscles. smooth muscles. Okay. So please remember this mnemonic, very, very easy, but definitely it is going to give 1 or 2 marks extra. You are going to solve these questions also, right? Usually these questions are asked in the AIMS examination, right? And since the quiz is also coming, so you might not know which question will fall into the quiz, right? So let's move on forward. This is the actin myosin we were talking about yesterday also, that this is the actin, which is the thin filament, and this is the thick filament. They do want to meet, but who comes in between? The papaji of myosin, that is the tropomyosin, which is the inhibitory protein, which is going to regulate that they should not meet without any reason or unnecessarily. The muscle should not contract when it need not to contract. Okay, so who is going to regulate the contraction and relaxation? The regulatory protein is going to be tropomyosin. To help that, we do have troponin I also, right, which is going to cause the inhibition of the actin meeting with the myosin. Okay, so yesterday also we have talked about it. Just to recap, that this is the papaji of myosin which regulates. Okay, now see over here, I told you yesterday also that these dark proteins, they are the active site of actin where the myosin can bind. Myosin cannot bind anywhere than these sites, but these are covered by these tropomyosin. So that's why the myosin head cannot bind to this actin molecules, right? There are 2 types of actin. Remember that there are going to be 2 types of actin. What are the 2 types? This round structure, they are known as G actin. And as soon as they form the filament from this G actin, what is G? G means globular. They looks globular in structure, so that's why these proteins are called as G actin. On the other hand, when they form the filament, this is known as filamentous actin. That is the F actin. Filamentous actin or F actin. Okay, so these globular molecule ultimately are going to form the filament, that is filamentous actin, on which this tropomyosin is going to be there. To help this, we do have to help or regulate whenever we want the contraction. The contraction should happen, right? We do have these pink molecule you can see over here. These are nothing but the troponin. The troponin alone, if it is there, the I and the T molecules, they will be activated. I told you yesterday also that this is the heterotrimeric protein, right? TIC, troponin. Troponin T is going to hold on to the tropomyosin, T for tropomyosin, I for inhibitory protein, and that inhibits the contraction, right? And C for calcium. As soon as the calcium is going to come from the this RYR and DHPR in the case of cardiac muscle, and from the RYR only in the case of skeletal muscle, this troponin C will be activated. As soon as troponin C is activated, it is going to drag that tropomyosin away from the active site of actin. It is going to act as the myosin's friend who can distract the papaji for some time so that the actin myosin can meet, right? So who is creating the distraction? This troponin C with the calcium. They both are distracting the papaji, right? So troponin plus calcium, when they bind together, they are making the troponin complex or troponin calcium complex. Calcium troponin complex is the better terminology. Complex, and that is going to distract the papaji, isn't it? As soon as the papaji are distracted, the actin and myosin, there is no one in between, they are going to meet together. And as soon as they are going to meet together, the contraction will take place, isn't it? So that's how the contraction is going to take place. Alright, so this is how the contraction is going to happen. You can see over here, the myosin, they are the thick filaments which are going to have head-like structure, right? And this is the tail part of it, okay? The tail of the myosin. And this is known as neck part, okay? So there are 3 parts. I'll just write it over here. What are the 3 parts? Just I told you that there is going to be head, bigger round head, and the bending part, wherever it is going to bend like this, right? So that is known as neck part of it. And the whole other part which is not going to be bending, it is present in that thick molecule, that is the tail, myosin tail. So there are 3 parts: head, neck and tail. Remember, in the head we do have ATPase also. So ATP needs to get hydrolyzed, right? For that we require ATPase, and in the ATPase what is going to happen? Definitely they are going to break down the ATP into ADP and PI. The phosphate, inorganic phosphate, that is going to give the energy. As soon as it is going to break down from the ATP, it is going to form the ADP and PI. That is going to give us the energy for the contraction, isn't it? Why the energy is required? Because we have to act against the gravity, right? So let's move on. So that is how the contraction is going to occur. First of all, let's see how the changes are going to happen in the actin and myosin, and there are different bands, there are different zones in which the changes are going to take place. And as soon as these changes are going to take place, the contraction will happen. First of all, we'll complete the contraction and then we will move on to the relaxation part. Alright, so let's see how the contraction takes place over here. So I'm going to draw the structure a little bit bigger, thicker is going to be myosin. So suppose this is the myosin fiber which is going to be thicker. And as you must have seen the golf ball rod, they do have heads, right? If I put a lot of golf sticks over here, then they will look something like this. Suppose these are the head of the myosin molecules. Neck and head part. Right, now what is going to happen? In between there is a part where these heads are not present. Tell me what it is called as. The chocolate question. This part specifically called as something. This is chocolate question. I want you to answer, guys. This part is known as. I'm asking you right now because there is a reason for it. Okay, this is the question for you guys. Okay, I'm waiting for the answers. Now I'm going to draw the actin molecule. So these are the thin filaments which are going to present like this. So these are the actin molecules. Okay, now what is going to happen? These are the actin molecules which are thin molecules. Some part of them are covered with the myosin and some is not covered. That means that is nothing but your overlapping and unoverlapped part, isn't it? So let's see what are these zones and bands are called as. Roy, think a little bit more, okay? Because I have asked this question. So Roy, this over here, the part which is non-overlapped by the actin, this is known as H zone. The heads are present or not present, I'm not concerned about it, okay? So I'll just, to clear your confusion, I'll draw like this. So a head part is still there, okay? The middle part where the heads are not there or the necks are not there, this part, this is my question. You are telling me you're also right, a bit clear, near to this answer, but not exactly right. This is the H zone. H zone where the myosin, only myosin is present. Whether the heads of the myosin, necks are present or not, doesn't matter, right? So only myosin. That means it is unoverlapped myosin part. Even if the heads are present, that is not going to bother us, okay? Now, I'm still waiting for your answer. Give me the answer. If you will not get to the answer, I'll give you ultimately at the end, okay? Now listen, what are the other part? Let's see. First of all, that this is the actin. This is the myosin. The whole myosin, whether it is overlapped or non-overlapped, the whole of the myosin, it is known as A band. So the red one is going to be A band. The whole of the myosin is known as A band, okay? And if I talk about the actin, actin is overlapped by the myosin and there is some part which is left by the myosin. Now if I take the unoverlapped part of this, let's say in the pink color, I'll write over here. So this is the unoverlapped part, unoverlapped actin, part of the actin or unoverlapped actin. This is known as I band. I band. So please remember, this is very, very important that this should be unoverlapped or non-overlapped part, okay? So this is known as I band. If it is covered by the myosin, I'm not going to include that, okay? If I draw imaginary line over here, in between, exactly in the middle of this I band. Okay, so suppose this is the I band. I draw the imaginary line over here. This line will be called as Z line, right? In the middle of I band. In the middle of I band, it is going to be Z line. So same Z line will be present over here also. So this is the I band, right? And the I band in between or in the middle of the I band, we are going to have Z line. There is one more line which is going to be present exactly in the middle of the myosin also, exactly in the center. That is known as middle, M for middle and M for M line. M line. So exactly in the middle we are going to have M line. Okay, so there are two lines over here. The zone that you told me, it is the H zone. And remember, whereas the neck and head is not present, empty part is H zone. Okay, M band. Okay, M band, there is no M band, which M line is there in the middle. Roy, you're right, but it is not the empty part, right? Now remember, this is known as, this is a chocolate question, this is known as pseudo H zone. Okay, so this part over here that I asked you, where the heads are not present, so this part over here is known as pseudo H zone. Nobody is going to ask you, but in the MCQs, for that purposes, you should be remembering that, right? So it is known as pseudo H zone. It is going to be bit lighter than the other part of, even it is not overlapped by the actin, but there are heads which are going to give a little, darker shade to that H zone, right? In the middle we do have lighter shade. As you come from the periphery towards the center, you are going to have the very light color shade in between, that is known as the pseudo H zone. Okay, now you are not going to forget that, right? If I would have just told you like that, then you would have forgotten it, right? Pseudo H zone. All right, so these are the bands and zones which are going to be present. A band, myosin, right? So let's revise that quickly. So A band, so I'm just going to write it in the write-up format so that this is not going to be confusing. A band, that is going to be whole of the myosin. Overlapped, non-overlapped, doesn't matter, right? I'm writing in the thicker font because it is the thick molecule. I band is going to be only actin. Should not be overlapped. Non-overlapped part. Non-overlapped by myosin. In the middle of I zone, in the center of I band, there is going to be a line which is known as the Z line. And from one Z line to adjacent Z line, what it is called as is known as sarcomere, right? The whole part from here to here is known as sarcomere. Okay, so this part from this line to this line, this is known as sarcomere. Sarcomere. From one Z line to another Z line, this is known as sarcomere, right? Z to Z line. Very good, Roy. So this is Z line to Z line, and then in the center of sarcomere, exactly in the middle, we do have, or we can say in the middle of the sarcomere, we do have M line. M line, right? And where the heads and necks of the myosin, they are not present, that is known as the pseudo H zone. And what is H zone? Non-overlap part, or I'm just writing only myosin. Overlapped plus non-overlapped over here. Overlapped or non-overlapped, both are going to combinedly form the A band, and only myosin, not overlapped by actin. If you are clear with these things, let's move on forward. Let's see in the contraction what is going to happen, right? So see over here, this is the Z line is written as Z disc over here, and what I was telling you that this from one Z line to another Z line, this is going to be whole of the sarcomere. My question to you guys, what is the length of the sarcomere, optimum length of the sarcomere? That means in the resting condition, what is the length of the sarcomere? Chalo batao fatafat se. This is your question, very easy question. What is the optimum length, or what is the length of the sarcomere? Now till that point we will discuss what is going to happen in the contraction. Contraction means my Z lines are going to come near, right? So you can see over here also that these are going to contract. That means the actin and myosin, the myosin heads are present, they are going to make the actin slide on them, and their heads are present on both the sides. So that's why both the actins from this side also and from this side also, they are going to come together or nearby. What is going to happen to the H zone? H zone will, Kamlesh, absolutely wonderful bache, tell me the unit also, right? 2 to 2.2 kilogram, meter, liter, what it is, right? You have to tell me that. Okay, half mark, otherwise it will be cut in the profs. Okay, so only half chocolate will be given. So what is going to happen? The heads of the myosin, they are going to drag the actin, and it looks as if the actins are sliding onto the myosin, and that's why this theory is known as sliding filament theory, which is given by the scientists, very very important scientists, the whole and soul respect for the scientists. You remember your name or do not remember your name, just remember all the name of the scientists, huge respect for them. Okay, yes, Kamlesh, now whole chocolate is yours. Okay, so 2 to 2.2 micrometers. Okay, micrometers, not millimeter. Millimeter is too much, you know. It's only micrometer, and that's why we require microscope. Roy, change your answer. It's not millimeter, bache. It's micrometer. Okay, then what is going to happen? So 2 to 2.2 micrometer. This can be seen only under the microscope and not like that. 2.2 millimeter hota to we could have seen it with the naked eye also. Okay, now listen to me right now. So myosin is going to make the actin slide onto it. I want the name of the scientist. I told you yesterday also. It's a very very important hint is H. It starts with the H. Okay, so you have to tell me. Now what is going to happen to the different zones and bands as the actin sliding onto the myosin? That means most of the part will be, more part of the actin will be overlapped by the myosin. That means the non-overlap part of the actin is going to reduce, and that means the I band is going to reduce. So the I band is going to reduce, that is for sure. You can see over here the I band here and the I band over here, they have, it is significantly reduced. So reduction in the I band which is seen, right? What is going to happen to the H zone? H zone is going to reduce, and ultimately when there is a complete contraction, actin are going to come together, it may disappear also. Okay, so what is going to happen to the H zone? In the complete contraction, it is going to be disappearing. So it disappears. It disappears also. And what is going to happen to the A band? A band and M line, they are very very solid. Remember, nothing will happen, no change. There are only two things, there is no change is going to happen, right? And yes, correct. So Roy, you are right. So what is going to happen? There are two things. Even if you remember all the things, you cannot remember all the things. Please at least remember these two things, that the A band and M line. How to remember? Ma never changes, right? Your emotion of your mother is never going to change towards you. No matter however you behave, your mom never going to leave you. So Ma, that means M line and the A band, they will never change. Okay, they will always love you. So Ma will never change. Remember that the M line and their A band, they will never change. Okay, their length is going to be similar. The M line is not going to change. It's a line in between, that is going to be the center part. What is going to happen to the Z line? Z line also comes closer to each other, so it shortens. the distance between Z line is going to shorten up, right? So distance between two Z lines, Z to Z line, it is going to shorten up, and that's why the whole of the sarcomere length is going to reduce, isn't it? So that is going to be the change. So I want you to remember that the M line and A band doesn't change, and all other almost reduce. The disappearance of one thing that is going to happen, that is the H zone. Okay, so with that, let's just write it down that Ma never changes, that is M line and A band, there is no change. H band, oh sorry, H zone is going to disappear. And the Z line, the distance between adjacent Z line that is the distance of, or the length of sarcomere is going to decrease, is going to reduce. All right, and then comes our I band is going to decrease because the I band shortens up, right? So suppose this is the resting length and this is the length at the time of contraction. What is going to happen? The first of all Z was here, but later on it moves here, right? So what is going to happen? Definitely, see over here, I'll draw it like this. So the Z line, the distance between the Z line is going to reduce. So I'm A band, I'm drawing on both the sides. So this was the A band, I band. This was the I band and the Z line in between. So this was the Z line, now this is the Z line. So this is the sarcomere length which is going to reduce, and in between we are going to have a thicker band that is nothing but the myosin. Okay, so rather than overlapping part, I'll show the myosin here. Okay, that is going to be the myosin whole structure. Now there is something known as phospholambin. Phospholambin, I didn't get the name of the scientist yet, so before going forward I'll just mention the name. That is the Andrew Fielding Huxley. The question came on this, so important. Andrew Fielding Huxley, Huxley and Huxley's sliding filament theory, it is famous, right? So please remember that sliding filament theory is going to is given by this Huxley scientist. Phospholambin, they are very, very important. Now remember, I told you in the sarcoplasmic reticulum, the whole of the calcium is coming from the sarcoplasmic reticulum in the skeletal muscle, isn't it? But at the time of relaxation, what should happen? During relaxation, the calcium, especially in the skeletal muscle, in the cardiac muscle, we do have two things that is required. We have to send back the calcium into the sarcoplasmic reticulum, as well as we have to send back the calcium into the ECF. So please remember, just now let's talk about the skeletal muscle. The skeletal muscle, the calcium should go back into the, for the relaxation, calcium should go back into sarcoplasmic reticulum, isn't it? And who is going to send the calcium back into the sarcoplasmic reticulum? That is going to be calcium ATPase, or it is known as the sarcoendoplasmic reticulum calcium ATPase. That is nothing but the SERCA pump. Remember, this phospholambin control or regulates the SERCA pump. How? It will inhibit it. Phospholambin is going to inhibit the SERCA pump. Remember, whenever your sympathetic system is active, what is going to happen to the SERCA pump? So whenever your sympathetic system is active, it causes the phosphorylation of phospholambin. That means the phospholambin will be inhibited, right? And if it is inhibited, that means there is no inhibition whatsoever whenever the sympathetic system is active. and that means there is no inhibition of calcium ATPase. The calcium, the duration that is required for the calcium to go back into the sarcoplasmic, what is going to happen to that? It is going to be decreasing. In less time, the calcium ATPase is going to get more and more activated because there is no negative regulation on the Cerca pump. So that's why it is going to cause the faster exit of the calcium from the sarcoplasm back into the sarcoplasmic reticulum, isn't it? And because of that, the calcium will not be available for the contraction in the sarcoplasm, and the relaxation will be very, very early as compared to the non-activated state of the sympathetic system. So in the sympathetic system activation, there will be faster relaxation which can be seen. In the less duration, the calcium will go back into the sarcoplasmic reticulum, and this phenomena is known as lusitropic effect. Please make a note of it. On the lusitropic effect, the short note usually comes. What is the luci? Luci means relaxation. How the relaxation is going to happen? We have to send the calcium back into the sarcoplasmic reticulum, right? Yes, it makes the muscle ready for the next contraction. For that, relaxation is important, so it is going to send the calcium very, very sooner into the sarcoplasmic reticulum. So normally the phospholamban, it has, what is the function of phospholamban? It is having negative regulation. That means it inhibits the Cerca pump in the normal resting condition, right? But in the case of sympathetic activation, there will be phosphorylation of phospholamban. Calcium ATPase will be activated more, right? And that's why the calcium will be going back into the sarcoplasmic reticulum very, very fastly. Okay, let's move on, and that's how the relaxation is taking place. Now, for the skeletal muscle, the calcium sources was the sarcoplasmic reticulum only, so we have to send back the calcium back into the sarcoplasmic reticulum, and that is going to happen with the help of this sarcoendoplasmic reticulum calcium ATPase, that is nothing but the Cerca pump, right? And then we talk about the cardiac muscle. Please remember there were two sources. Can you tell me how the ECF calcium will be sent back? I know you are going to answer me that the calcium source are from the sarcoplasmic reticulum also, right? And same over here, we are going to activate the Cerca pump, right? Why that it is going to, calcium is going to get returned back into, calcium is going to return back into their respective places, First is sarcoplasmic reticulum, second is this ECF, right? You're absolutely correct. With the help of sodium calcium exchanger, we have talked a lot about that in the negative the inhibition of this sodium potassium ATPase also. So there is a pump which is known as sodium potassium ATPase, right? Which is going to create the gradient inside the cell. It is going to throw out the sodium, more sodium outside. That means less sodium will be available inside the cell, and because of that there is the gradient which is going to get created. And once the gradient is created, which is going to be helpful for the next thing, that is sodium is going to enter, but we have to maintain the electrical balance also, and that's why one positive ion inside, one positive ion outside. Okay, so we are going to throw the calcium out, and with the help of this sodium calcium exchanger, this is the standard short form for this symporter, sodium calcium exchanger. With the help of that, we are going to send the calcium back, right? Yes, it is driven by the sodium potassium ATPase, and the drug that inhibits this, and the calcium will be present more inside the sarcoplasm, that is nothing but the digoxin digitalis, all these things. Okay, let's move on to the next thing, that how the ATP has the role in the cross bridge cycling. Okay, so we are going to learn the role of ATP role in the cross bridge cycling. Okay, role of ATP. What is the role of ATP? Now see, suppose this is the neck part of the myosin and this is the head part of the myosin, okay, and this is the actin over here. Now what is going to happen? The attachment of the ATP will cause the detachment of the ATP will cause the detachment of the myosin from the actin. That means it is responsible for the relaxation, right? So first thing first, that as soon as you can see over here, that as soon as the ATPs are attached, the molecules are going to detach. The myosin from the actin will be detached, okay. Now there is one fallacy in this, that the only way this myosin is going to move, that they have shown the movement like this and this, okay. So this is the movement which is possible in the myosin, right? So it is going to get detached. So if I'm talking about it, it is going to get detached. Now the ATP is still attached, right? It is going to get converted into ADP plus PI. As soon as it is converted into ADP plus PI, what is going to happen? See over here, as soon as it is converted into ADP plus PI, the molecule which were bent first. Now there are two things which are happening. Now this is the pivoted condition of the myosin, where the head and neck are bent, right? As soon as they come in the 90 degree, that is known as the cocking of the head. When the cocking of the head is going to happen, so this position is known as cocked up position. When there is going to be cocked up position of the myosin, when the ATP will be converted into ADP and plus PI, but they are still attached to the myosin head. Now what is going to happen? They are still attached, but the calcium, if it is not present, obviously you cannot remove the tropomyosin from the active site of actin, and that's why we require calcium over here. So calcium is required. As soon as the calcium is present, what is going to happen? There is going to be binding of this actin and myosin fibers that is going to take place. Myosin head will be attached to the active site of actin, okay. Now as soon as these two molecules will be detached from this, what is going to happen? The power stroke will be taken place, right? So as soon as these molecules will be removed, now the head will be pivoted back again towards the 45 degree angle, right? These 45 degree, 90 degree haven't given in the boron or in the Guyton and Genong, so that's why I'm not going to write it over here. And you can, if you want, you can write it also, that pivoted condition, that means 45 degree angle in the myosin head and the body, and the cocked up position, that means 90 degree angle around, okay, so perpendicular. So when they are going to get released from, they are going to detach from the myosin head, definitely it will be the pivoted condition which is going to be present. Next molecule of ATP is going to be joining. Now understand that when I said that this is going to be cocked up position which is going to be present, that means it is going to bring the actin. Now they are not detached with the actin. As soon as the ADP and PI they are detached, the actin, the cocking up will happen. But for the detachment of actin and myosin, we require ATP. ATP are less in the case of smooth muscle, and that's why it gives rise to one very specific phenomena that I want you to answer in the comment box. Till that point, we'll just write 3 points which will give you the role of the ATP. So first of all, binding of ATP to the myosin head. Binding of ATP to myosin head. What will happen? That will get, the myosin head will get detachment of myosin head from actin, right? Now this new molecule of ATP has bound just now, right? After that, what is going to happen? The ATP will be converted into ADP and PI, but these will be still attached to myosin, okay? Now what is going to happen? As soon as they are hydrolyzed, obviously there will be the cocked up position which is going to be present. You can see over here that this is going to be cocked up position which is going to be present. So which is going to be present. Now this ATP and PI, we have to remove that. Or let's say detached from the myosin. What is going to happen? As soon as they are detached, there will be pivoting. The myosin head will be pivoted. Dragging the actin in the center. From both the side, it is going to drag the actin in the center, and that's why the sliding filament theory stay put over here. Very good, Kamlesh. I got the answer from your side that this is going to lead to latch bridge mechanism, right? And this will give rise to sliding filament theory. It looks as if the actins, they are sliding onto the myosin, right, given by the A.F. Huxley. Now new ATP will be bound. Okay, I have written everything about the ATP and myosin in the black color. You have noticed that. So now new ATP will be bound to myosin, and then only the myosin head will be detached from actin, right? So this is going to happen and the cycle will repeat as many times the calcium is going to be present. So where we require the calcium, calcium is required when we have to attach the myosin, right? So cocked up position, if the calcium is present, so I'm writing over here, with the help of calcium, the actin will bind to, the myosin will bind to active site of actin. I'm just writing actin over here because I don't have space. So I'm going to write the actin over here. So this is important. Calcium is also important with the help of the hydrolysis of this ATP, right? So this is the whole condition which have shown in the diagram also, and we have written that, right? So let's move on to the next thing. Can you answer me? Can you answer me of this? Give me the answer of this question. Which of the following will be seen in the middle of H band in the sarcomere? H band itself in the middle. So in the middle of that, what we have? M line, Z line, A line, I line. Tell me the answer, A, B, C, D. You can see over here that the answer is not given. What I'm asking is, in the middle, what we have? Give me the answer of this. I'll move on till that time. I'll move on to the next thing, that is, don't miss out on something that could be great just because it also could be difficult, right? So no matter if it is difficult, no matter it is easy, the courage to continue, that counts. Okay, so this is a very wonderful message that we are having, and this background and everything made by myself, right? I'm very proud of it. Whenever I want to do time pass, I do such things, right? So the background, the gradations in the background, the color combination is given by me. So yes, you are right. Only one answer I got. Kamlesh, absolutely wonderful bacha. That is the M line. In the exactly middle, we do have M line over here, right? So we are going to have M line. Let's move on to the next thing. Okay. Good evening, Rahul. You're a bit late, bache, but I'm happy that you came, right? Let's move on to the next thing. What is the next thing? As we saw in the nerve, we are going to see in the muscle. I'm going to dissect the muscle. Okay, Roy, I got your answer now. So I'm going to dissect the muscle. I'm going to go inside, right? We are going to go on the levels of protein also. There are not just 4 protein that we have seen, actin, myosin, tropomyosin, troponin, but there are many molecules which are present, right? We haven't discussed the regulatory molecules. That we'll discuss. Now see over here. First of all, when the whole muscle I'm going to take, there is a chunk of muscle which is going to be present, right? How the muscle is formed, you know that whenever you go to the any gym, right, they ask you to purchase some things from them, right? Some powders, some formulas, right, which do have lot of protein in it. Why? Because we know that the muscles are hypertrophied with the help of protein. And why? Because they are made up of protein. So if you give more protein, you eat more protein, your muscle is going to grow, right? So same over here. Proteins come together, a lot of proteins come together, they form a single muscle fiber. Muscle fiber, which is known as myofibril. So see over here, this is known as myofibril. Such myofibril, many myofibril come together and they are going to form something known as fascicle, right? Now understand that if I say that many fascicle, that means group of myofibril, they are going to come together. What they are going to form? They are going to form the whole muscle, isn't it? But understand that they do have separate covering onto them. Okay, so the whole muscle is not shown over here, so I'm just highlighting it. So whole muscle will be formed. Okay, now understand the myofibril will be covered by the connective tissue layer or covering. Then the fascicle of the muscle will be covered, right? And the whole muscle will be also covered, which is known as sarcolemma, which is going to form the sarcolemma, right? The whole muscle is going to get covered. Now, what are the things? Myofibril is covered by what thing? Myofibril. Again, I'm going to tell you EP is the mnemonic, right? E is not followed by P, that means it is going to be the endo, and we are covering the muscle, that means it is the endomysium, isn't it? It is going to be the endomysium. Then fascicle, perimysium. As in the nerve we have learned about endoneurium, perineurium and epineurium. Same over here we are going to learn about the endomysium, perimysium and epimysium, right? And when we are going to learn about the series elastic protein and parallel elastic protein, please remember they are present parallelly to the muscle fibers. That's why, because these are the muscle fibers, let's suppose, right? And this is the whole connective tissue layer which is going to go parallelly onto it. So this is known as parallel elastic components. So all the covering, they come under parallel elastic component of the muscle. Series elastic component, see over here, to the series, in the series of muscle we do have tendons, right? So mainly the tendons are going to be coming under the series elastic component. When you are going to learn about the isometric and isotonic contraction, this information is going to be very helpful to you guys. Okay, let's move on ahead. That what are the sarcolemmal protein? Now understand, the sarcolemma is different, right? And the whole muscle is different. Sarcolemma is just the covering that we have talked about, right? So in the sarcolemma also we do have proteins, and in the muscle also we do have proteins. So what are the sarcolemmal protein? Very simple mnemonic I'm going to give you. There are 2 proteins which are present in the sarcolemma. We have divided that into integral protein and peripheral protein. Sarcolemma is the membrane, right? And we have learned in the general physiology itself that onto the membrane we do have either integral, that means transmembrane protein, and we do have peripheral protein. What do you mean by trans? Trans means through, right, which is going through and throughout of the membrane. So they are known as transmembrane protein, all right? So if they are going from outside towards inside, they are known as transmembrane protein. Peripheral, either they are only present on the outer border or inner border. This is known as the peripheral protein. How to remember which type of proteins are there in these 2 types? Just remember over here, in the integral we do have G, and in the peripheral we do have not only single P but double P over there. Okay, that's how I remember. You can remember the way you want. Okay, so just remember there are 2 protein. So glycans are going to come into the integral protein. Why? Because the G is present in the integral, right? So please remember over here there are two types of protein which are present. First is the sarcoglycan, and then comes the dystroglycan, right? And I told you P are there in the peripheral, so I'm going to write over here that first thing is syntrophin is going to be present and dystrophin is going to be present. Do you remember that we have talked about the dystrophin protein's mutation? Tell me, if the dystrophin protein is mutated, what disease you might have, or why you will have, your patient is going to have? Tell me the disease that your patient is going to have if the dystrophin protein is mutated. Tell me. Mutation. And there are going to be two conditions: partially mutated and completely mutated. You guys are alive till now? I'm waiting for the answer. Yes, please. There are not only single type, but two types of mutations are going to be there. What are the two types of mutation? If it is partially present, right, and it is completely absent, there are going to be two types of dystrophies which are going to be present. I want the answer. Till that time I'll wait. I'll move on forward. And if you do not give me the answer, obviously I have to give, right? So there are going to be muscle protein. Now I told you we have learned about sarcolemmal protein. That means what are the proteins which are present on the sarcolemma, on the covering. Let's go inside, dive inside into the muscle. What are the proteins that are required for the muscle itself? There are three types of proteins. As we divided two groups on the membrane, right, itself, that is integral and peripheral, we are going to divide functionally the muscle proteins as the contractile protein, which are going to help in the contraction itself, which is the main function. And we already discussed what are the proteins: actin and myosin, right? Actin and myosin, that is going to be there. What are the regulatory proteins? Who are regulating when the contraction should happen, when it should not happen? First was the Papaji, right? And that was regulating, negatively regulating it. So tropomyosin, and then comes our troponin to help that. Troponin is kind of mother, whenever it requires to take a side of dad, it will take the side of dad. That means at the time of relaxation, it will help tropomyosin also. But whenever the contraction is required, as far as the calcium comes, it is going to take the other side, that is the side of the actin. Okay, so troponin is the in-between kind of a regulatory protein. Okay, anchoring protein. There are some anchoring protein. Now remember, the sliding is occurring continuously. It might possible that the actin will move away from the Z line, or it will get detached from the myosin itself. It will go somewhere else. This may be possible. And to hold these actin, myosin, troponin, tropomyosin, all the fibers on the same length, same side, we should have, yes, I got the answer right now, Kamlesh. Okay, there may be some lag. This lag is very, very long, but yes, I got the answer from you. That is Duchenne muscular dystrophy and the Becker's muscular dystrophy, right? BMD and DMD. I'll write that. Coming to the anchoring protein, there are 6 proteins. Remember the name of them. That the first of all is going to be your desmin protein. Then comes the alpha-actinin. Then there is going to be one protein which is known as nebulin, right? Then the tropomodulin. It's not tropomyosin. Tropomyosin is the regulatory protein. Tropomodulin is going to be your anchoring protein, isn't it? Then there is tropomyosin, desmin, alpha-actinin, right? And then comes the titin, which is very, very, very important. Why? You'll just get to know now. Okay, titin is very very important. Why? Because it is maintaining the elasticity. And ये सबका बाप है. क्या है ये? It's the largest protein available. 30 lakh Dalton is the weight of this. Okay, so it is the biggest protein which is present in the muscle, in the human body, which is present in the muscle. Yes, cardiomyopathy will come on to that. There are different types of cardiomyopathy which is going to happen due to the mutation of these anchoring proteins. Okay, we'll come on to that. Yes, myopathy is also possible because of the titin. Okay, coming to the partial absent and complete absent. Partially, if it is present, remember it is known as BMD, that is Becker's muscular dystrophy, and it is completely absent. This is known as DMD, that is Duchenne muscular dystrophy. So if it is partially present, Becker's muscular dystrophy, बेकार है, right? It's बेकार. This dystrophin is present, but बेकार dystrophin is present, right? It is not completely present. So remember बेकार, that is Becker's muscular dystrophy, but it is completely absent. It's gone, right? दूर दूर तक कहीं नहीं दिखाई दे रहा. So that is going to be your DMD, that is Duchenne muscular dystrophy. Okay, and one sign is going to come into the Duchenne muscular dystrophy specifically because the muscles are not having this sarcolemmal protein, which is important to hold on to the actin. So sarcolemma to the actin, it is going to connect it. Okay, so the dystrophin is going to connect from the sarcolemma towards the actin. The actin molecule will be hold on to that position because of this dystrophin. If that is not there, there will be not proper force which is going to get generated, and that's why whenever a child is going to, because it's a genetic mutation, so it can be seen in, it is going to be seen in the children. So the child is going to walk on his own body. He's going to take the support of his bones and he is going to change the position. And this is known as dash dash sign. It is known as Gover's sign, okay, which is going to be seen. It looks like as if the child is climbing on his own body to stand up from the sitting position. Why? Because there is not enough force which is going to get generated because of this mutation, right? So this is the condition which is going to happen. Let's discuss this anchoring protein and then we'll come back to the Duchenne muscular and Gover sign. I do have slide for that. So first of all, we'll discuss one by one desmin. Remember, the desmin is going to connect the sarcolemma to the Z line. Okay, so it is the connection of sarcolemma to Z line. Okay, what they are going to do, they are going to keep the whole sarcomere intact and they are going to maintain their length. Okay, so that is the function. Alpha actinin, which is also known as cap Z. So it is like a cap on the actin. So please remember, it is like a cap on the actin. So where it is going to be present? It is going to be present on the Z line. That's why it is known as cap Z, right? So it is present on the Z line and as if it is put on the cap on the actin. So definitely it is going to be present in between. Yes, please. It is going to connect the actin and stay put the actin connected to the Z line, right? Stay put connected to the Z line. So the alpha actinin will be connected to the Z line and to the actin on the other side, right? Then comes the alpha actinin nebulin. Nebulin, where it is going to be present? Nebulin is going to be present in between. Suppose this is the Z line, right? And this was the actin. Over here, we are having the cap Z. And within the actin, we do have what thing? From the one end of the actin towards the Z line, through the actin, we do have the nebulin protein. Okay, so nebulin protein is going to be present throughout that I band. Okay, and what this nebulin protein is going to do? The nebulin protein maintains the length of actin. Through the actin, what is present through the actin? Suppose this is the myosin. Okay, so through the actin, the whole actin, we are going to have this nebulin protein, which is going to maintain the length of this actin. Okay, now I told you that let's say this is the pink molecule, which is the alpha actinin or CAPZ. Now on this side of on the M line, exactly to be precise, on the M line we do have another molecule on the actin that is known as the tropomodulin. Okay, so tropomodulin is going to be present on the other side of the actin, and that is also helping to maintain the actin's length. Okay, so tropomodulin is going to do the same thing on the myosin also. Myosin will be connected to the M line and double M are coming over here. So please remember, this is nothing but the myomesin. It will connect the myosin. Myosin itself, myo is there in the name itself. Myosin will be connected to the M line. Right? That is the function. So the central position of the myosin is always maintained. Why? Thanks to this myomesin. Okay, so we have done with the desmin, alpha actinin, nebulin, and you were talking about the cardiomyopathy. So I'll just mention over here that if these two, any of these two are defective, what is going to happen? Please remember, if any of these two are defective, then there is going to be a myopathy, cardiomyopathy, which is going to be there. That is known as Neem line cardiomyopathy. Neem line cardiomyopathy. Okay, Neem line cardiomyopathy. Okay, which is the most commonest protein to be defective in this? That is going to be nebulin more than the alpha actinin, and that's why it comes from NN. It is easier to remember, Neem line cardiomyopathy. Okay, right. So let's move on to this. We have discussed the tropomodulin also and myomesin also. Let's discuss the next thing, that is nothing but the titin. Titin is known as the elastic spring of the muscle. It is known as the spring of the muscle, which gives the elasticity. Spring of the muscle. What it is going to connect? It is going to connect the M line to Z line. It is going to connect the M line to the Z line. Now, there are two types of cardiomyopathies which are present, or there are two types of mutations which are going to be present also. Okay, so there are two types of cardiomyopathy. Can you tell me what are the cardiomyopathies? Yes, please. Roy, Kamlesh, answer karo bacho. What are the two cardiomyopathies if it is defective or mutations? Let's write mutation of this titin can give us the cardiomyopathies. What are the cardiomyopathies which are going to be present? Yes, please answer do bacho. I'm waiting for that. And there is one more thing which can be possible, and what is that? That is nothing but the LGMD, that is limb girdle muscle dystrophy. Limb girdle muscle dystrophy, which is going to be present in this. Okay, so I'm waiting for the answers. X link, Y link nahi bache. Tell me the name of cardiomyopathy. Yes, please. Cardiomyopathies that are present, that is, first is hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy and dilated cardiomyopathy. Both can be possible. I'm just writing CM over here. That is cardiomyopathies which is going to be present. And limb girdle muscle dystrophy, that is most commonly because of this. This question came as the NEET question in the 2020 itself. So please remember, limb girdle muscle dystrophy, most commonly affected protein is going to be titin. Okay, and hypertrophic and dilated cardiomyopathy, that is also going to be titin. We have discussed about Sarka pump, and if that pump is defective, what is going to happen? If just a bit of information. Sarca pump, if there is a mutation in that, then it is going to lead to Brody's myopathy. Now remember, it is not a muscle protein. I'm just writing over here because we were discussing about the myopathies. We have discussed nameline myopathy. We have discussed the limb girdle, this limb girdle muscle dystrophy. Then we have discussed about dystrophin also, that is Becker's and Duchenne's. We have discussed about dilated hypertrophic, and that's why I have written over here the Brody's myopathy. Now understand, in this, Sarca pump is mutated. What was the function of Sarca? The function of Sarca was the relaxation of muscle. Will the relaxation possible in this? No, it is going to be very, very delayed, and that's why the patient is going to have the symptoms like prolonged muscle contraction, like spasms, cramps, and they're going to have wrinkling of the face very, very easily, right? So yes, concentric myopathy, yes, you are right. There are two types of hypertrophies possible, concentric and the eccentric. In the concentric myopathy, what is going to happen? The lumen will be shorter, and in the eccentric, one type of hypertrophy is going to happen, that in the lumen is going to be same, but the muscle is going to grow. Okay, yes, you are absolutely correct, Roy. So that is going to be there. What the other students are doing, guys, just participate in the this discussion. That is going to be very, very interesting for us, right? So there is a difficulty in muscle relaxation. Muscle relaxation, which is going to be there, which is in the Brody's myopathy. The patient will come to you as cramps and spasms of the muscle, and if you see prematurely, there will be wrinkles. What are the wrinkles, by the way? The relaxation won't happen properly, and that's why the wrinkles will come. The contraction are going to be there, right? That is going to be wrinkle, and that's why we give Botox. What the Botox is going to do? It will cause the relaxation of the muscle. It won't allow the muscle to contract. Okay, and the minute amount is given, minute dose is given. If it is more, the muscle won't be able to contract. And why? If the muscle, let's say the toxicity happens in the blood, the respiratory muscles won't be able to contract, and the person can die. Okay, so that's why it is the toxin. Okay, Ronit Verma, good evening. No issues, bache. That will be there for some days. We are going to keep that on the YouTube, and definitely after some time it will be there in the app. Okay. Yes, corrugated facial expressions are going to be there, right? Let's move on to the next thing. What is going to happen to the next thing? Let's move on. Dystrophin we have already discussed, mutated or partially present. If completely absent, the sign I was telling you was the Gover's sign. He was the scientist who discovered it. Partially present है तो BMD होगा, completely absent है तो DMD होगा, है ना? बेकार वाला डिस्ट्रोफिन अगर है, if the बेकार dystrophin is still present, that is known as BMD, Beckers muscular dystrophy, and दूर दूर तक कहीं दिखाई नहीं दे रहा, तो that is going to be Duchenne's muscular dystrophy. And this I was talking about. First thing, see on this right side over here, what you can see over here, that this is the sarcolemmas, right? Sarcolemma means the covering, and you can see over here that this is the actin, the red molecule that they have shown over here. This is the actin. So dystrophin is this blue one protein. What it is joining, see over here, from the sarcolemmal protein, it is joining or keeping the actin on its particular site. Okay, so that is the function. If dystrophin is not there, definitely the actin is continuously moving here and there, and that's why it is not able to have the proper cross bridge cycling, and that's why the proper force is not formed, and because of which the subject, or especially the children's, you can see he needs to stand up, but he's taking the support wherever it is possible, and after a point what is going to happen, he's going to start taking the support of his own body to stand up, right, which is looking as if he's crawling or he's climbing on his own body. Yes, there is going to be proximal muscle weakness also. So he's going to climb on his own body, and this is known as Gover's sign. go aside, right? Let's move on to the titan genetic mutation. We have talked about it, and please remember that this is the largest protein present. It is known as muscle spring. Largest protein present in the human body. 30 lakh Dalton is the weight, if you want to remember. And genetic mutation is going to cause two types of cardiomyopathies, that is dilated and hypertrophic, and limb girdle muscle dystrophy is also possible, right? Now coming to the another thing, that is, what if the antibodies are formed? There are two types of antibodies can be formed, pre-synaptically and post-synaptically, or we cannot say over here the synapse per se, but we can say over here pre-junctional and post-junctional. That is the actual correct terminology, because this is not the synapse. Synapse, we cannot use the terminology everywhere, but usually this is called as pre-synaptic or post-synaptic in all the books. So it is there, right? There are going to be antibodies which are going to form. You can see over here that there are channels which are present. First of all, there is a calcium channel, and this is the nerve terminal, axon nerve terminal. You can see that this is the axon nerve terminal. So it is important for exocytosis. If the antibodies are formed against it, what is going to happen? This channel won't be able to work. That means the calcium is not getting the entry inside the nerve terminal, and if it is not getting entry, what is going to happen? There is no one to scold us to come on the weekend and take the class, right? So what is going to happen? There will be no exocytosis. We will be lying down and enjoying our comfort zone at room, right? Hostel room or wherever. So that is the thing which is going to happen with the neurotransmitter, isn't it? So the neurotransmitter won't leave the comfort zone. They will be there inside the vesicle, right? So that means there will be no exocytosis. No exocytosis, that means no contraction is going to be possible, and that syndrome is known as Lambert-Eaton. I should have asked you, but anyways, right? Then there is another most common out of these three, that is myasthenia gravis, and the actor Amitabh Bachchan, he is having this myasthenia gravis, right? So what is going to happen in this? First of all, when we learned about Lambert-Eaton, the muscle is not able to contract, okay? Later on, we have moved on to the myasthenia gravis. See over here, the problem is in the receptors, okay? What is going to happen over here? The problem is that the receptors will be blocked by these antibodies. There is no problem in the exocytosis. The exocytosis is happening, but the neurotransmitter, they are not able to go and sit on the receptors, right? It is like a musical chair. You can just correlate with that there are limited numbers of seats, but there are more numbers of candidates, okay? So what is going to happen? Who is going to win? Whomsoever is more in concentration. So if the antibodies are more in concentration, they are going to win the game, right? If our neurotransmitter are higher, they are going to win the game. So they are going to sit on the receptor. Now understand, when we wake up from the sleep in the morning, what is going to happen? Our neurotransmitters are ready in the vesicles, right? So there is a store of neurotransmitter ready to get released into the neuromuscular junction. So what is going to happen in the myasthenia gravis, specifically in the morning? Yes, please. The neurotransmitter levels are much higher in the morning, and that's why the muscle will contract. But as the day progresses, in the evening, this neurotransmitter level won't be able to synthesize at that rate, and that's why the level of the neurotransmitter start decreasing. But the level of antibodies, they won't change. It is still that much, but the neurotransmitter levels are decreasing, and because of which automatically or relatively the antibodies are going to have more concentration as the day progresses, and now they will start to win the game. Why? Because their concentration is higher now in the evening, so they are going to sit on that receptor, and that's why the neurotransmitter cannot sit onto it. And if they are not sitting onto it, obviously again the sodium channels won't be able to get open, and the depolarization won't happen. The whole contraction will not take place, and that's why in the evening they are going to have worsening of the symptoms. Why? Because the neurotransmitter synthesis will start to decrease. In the case of Lambert-Eaton, what is going to happen? Understand, this is present in the you can say against the calcium. So as soon as you give more and more stimulus, it depends on the stimulus. What happens is that the calcium channels have tentatively more probability to open up if you give more stimulation. Now understand, if you give first stimulus, second stimulus, third stimulus, fourth stimulus, the probability continuously is increasing to open up the calcium channel. The calcium will start accumulating outside these channels, and their concentration is going to overpower the antibodies. Right, they will fight with the antibodies and they are going to open up the channel. As soon as the channel, they are going to remove those antibodies aside and they are going to get inside the nerve terminal. That means what is going to happen in the Lambert-Eaton syndrome, that the patient will see as the day progresses towards the evening, it is much more easier to contract the muscle. First of all, in the morning, he is going to have the problems to contract the muscle. Why? Because we are not able to give that many stimulus, right? We have to give more stimulus to the nerve. More the stimulus, it is going to be easier. So I always tell you the initiation is difficult, right? And the later on part, as the momentum grabs up, the calcium start to enter inside the nerve terminal and it is going to be, the exocytosis will happen. Okay, so these are the major differences between these two things. So please remember, first of all, the Lambert-Eaton syndrome, and second is the myasthenia gravis. Third, where we'll talk about it, right? So first of all, we'll write that thing. So first of all, Lambert-Eaton syndrome and myasthenia gravis. Lambert-Eaton, the antibodies are against the calcium channel, voltage gated calcium channel. I'm writing V over here, in nerve terminal or axon terminal, you can write nerve terminal or axon terminal. In the myasthenia gravis, it is against the acetylcholine receptor on the sarcolemma or motor end plate. You can write motor end plate also. In the evening, as the day progresses, the symptoms will get worse, that is worse, symptoms are going to get worse, worsening of is seen, and as the day progresses over here, myasthenia gravis mein, what is going to, the symptoms are going to get better, relieve the symptoms are relieved, so the patient will get better, rather the condition is going to get better. Rather than writing symptoms can increase, the condition, let's write condition, is going to get better in this scenario. What about this? As the day progresses, the condition is going to get worse, right? So these are the major differences. This is present on the nerve terminal, right? That means before the neuromuscular junction, and this is present post junctional, that means on the motor end plate. So these are the major the changes or differentiation point. So how to remember? Suppose if I give a food, suppose this is the table, right? Suppose this is the table onto which there are two sides of it, right? One subject of Lambert-Eaton syndrome and one subject of myasthenia gravis is present. I don't know why it is not visible completely, so I hope I have drawn this type of diagram. So myasthenia gravis, you know. So what is going to happen? I gave the food to both of them. On the weekend, I called asked them to come to my home and I gave the food, right? There is a party. They are just the guinea pigs for me, right? So I gave the food whatever they want. Now what is going to happen? First of all, the myasthenia gravis patient, he will have no problem in eating. Why? Because the condition is going to get worse later on as he is going to use his muscle. So first of all, he will start eating the food, right? But later on, he is going to have the weakness in the muscles, so he won't be able to to eat the food later on. But first of all, he will start to use. So who is going to eat first? The myasthenia gravis is going to able to eat the food first. But if I ask the in the case of Lambert-Eaton, what is going to happen? The name is Eaton, but he is not able to eat the food. Why? Because there is not enough stimulus in the beginning so as to open up the calcium channel. But later on, he tries very hard. As soon as he sees that the other person is eating up everything and he is not able to eat, he gathers the courage, right? He stimulates his nerve so many times so that later on, after some time, he is able to eat the food. Okay, but till that time the myasthenia gravis won't be able to eat the food later on. Okay, so who will start later? The Lambert-Eaton. He can eat till the end because he'll have the problem in the starting, but later on he's going to eat the food. Okay, so that's how I used to remember. You can remember the way you want, but it is going to be helpful for you guys. Okay, chalo, so this is the diagram that I have drawn, right? It's the weekend. I hope galti maaf hogi, right? So these were the differences between the skeletal muscle and the cardiac muscle. And there are different antibodies. Let's talk about this Isaac syndrome. Remember, this was the question came in 2018 AIIMS exam. So I have put it on this. So Isaac syndrome, what is Isaac syndrome? Please remember, the Isaac syndrome is the thing in which the antibodies are formed against the voltage gated potassium channels. As we do have sodium channels and potassium channels also there on the nerve. Why? Because for the depolarization and repolarization, it is required. Repolarization will be followed by the relaxation, and for the relaxation we require potassium to go out. है ना, टाटा बाय बाय करना पड़ेगा पोटेशियम को, otherwise the depolarization will stay there. There will be no repolarization. Right, and if there is no repolarization, the next stimulus cannot come. That's the problem. So we want the potassium to move out. But if the antibodies are there are guards present who are not letting the potassium to go out, what is going to happen? Definitely the relaxation won't take place, na? The repolarization won't take place. So what is going to happen? In the initial case, there is continuous depolarization which is coming from the sodium and the calcium, and there is no depolarization. Remember, I'm telling about the initial phase. So what is going to happen? Definitely there will be a lot of neurotransmitter release which is going to take place, and the patient will feel the symptoms like cramps and sustained contractions, right? Thankfully, this is going to affect only and only peripheral nervous system, and thankfully it won't affect our smooth muscles. Otherwise, please remember, the viscera contains smooth muscle. All of the time your gallbladder will be contracted, all of the time your intestine will be contracted, all of the time your urinary bladder will be contracted. Imagine, we have to live in the washroom, right? So that is not going to happen. Thankfully, the nature has given this thing or this disease or lesion to the skeletal muscle only. Okay, so this is mainly seen in the skeletal muscle. Skeletal muscle, and seen in the peripheral nervous system. Okay, so these are the 2 points that you have to remember. As soon as we are going to move on forward, you will get to know. In the myasthenia gravis, please remember, the most commonly the antibodies are formed against, there are 2 types of acetylcholine receptors present depending on the protein molecules and what are the changes are there. Okay, so first is the receptors, that is acetylcholine, and there are on the sarcolemma we do have another protein which is known as musk protein. So this is going to be the second most commonly affected protein which is going to be get affected in the myasthenia gravis. Okay, if the acetylcholine receptors are not in the option, then only you are going to move ahead with the musk protein, right? Otherwise you won't be moving. Till now everybody understood everything? Give me a thumbs up or something. Do you understand the Isaac protein? Yes, bacho. Do you understand the Isaac protein, this syndrome? Let's move on. If you understood this, let's move on to the skeletal muscle and smooth muscle. What are the differences? These are the differences that you have to enlist. Okay, first of all, what is the shape of skeletal muscle? All know it is kind of a cylindrical shape. Okay, there is a long muscle which is going to be present, and if you see in the resting phase, it is going to be kind of a cylindrical shape. Okay, so I'm going to write over here. The shape is cylindrical. On the other hand, if you see the smooth muscles, you'll see that they are very, very short, and the belly of that muscle is going to be widened up in between, right? So Kamlesh, on what you are laughing, I don't know. एक तो lag इतना है इसमें. Anyways, so shape of the smooth muscle is going to be spindle shape. Alright? What about the nuclei? Remember, the skeletal muscle has to get repaired many a times, and you must have seen that if you eat protein, your smooth muscles are not going to grow, but your skeletal muscles are going to grow very, very rapidly as compared to the smooth muscle. Why is it so? Whenever you're doing exercise, that means strength training, what you're actually doing is you're causing the micro tears in the skeletal muscle. But you must have seen that you are going to have a lot of soreness, you want to impress someone, so you join a gym and you start doing or putting up start doing the gym with the very heavy weights. What is going to happen? Your muscles are going to get sore. Why? Because there are so many micro tears at the same time which are going to happen. No issues. It's funny, right? So what is going to happen? Definitely there are going to be so many micro tears that actual tears will start coming, right? So then what is going to happen after 2 to 3 days? Your muscle will start getting repaired, and that's why you are going to have less pain, right? Why there is a soreness or pain? Because the inflammatory mediators, they are going to get released because of these micro tears. Why is it so that the repair of the smooth muscle is prolonged, or it is going to take a lot of duration as compared to the skeletal muscle? Within 1 or 2 days, you are going to have hypertrophied muscle, toned muscle, right? And you're going to have the repaired muscle back to the use or abuse, right? So remember, because they do have very good genetic material, because they are multinucleated. Let's keep the color same. So many nuclei are present in a single muscle, and that's why their repair is going to be faster. Obviously, the nucleus is going to be the factory of all the molecules, right? The coding, genetic coding is present in that nucleus. So that's why we require a nucleus so as to produce the growth factors and other things, right? What about the smooth muscle? They have single nucleus. And that's why what is going to happen over here? Definitely, if they have single nucleus, their repair is going to be a bit delayed, right? What about the control? You can control your skeletal muscle, right? As I'm talking non-stop since 6 o'clock. So control. So it is going to be voluntary. This is going to be involuntary. Involuntary. What about the troponin, titin, nebulin? Were they present in the skeletal muscle? Yes, of course, right? They were present, and we have talked about titin, especially a lot of time we have talked about it, right? So definitely the titin, they all are present in the skeletal muscle, but they, remember, are absent in the smooth muscle. Very, very important point. Just make a mention of this. Specifically, if I ask you, troponin is not present, then how the tropomyosin will be displaced? And the next question, is it the requirement to displace the tropomyosin in the smooth muscle? Yes or no? Give me the answer in the comments. Instead of troponin, there is a molecule which is going to be present, which is known as myosin light chain kinase. Okay, the kinases and phosphatases, they are going to be present in the smooth muscle, which are not there in the skeletal muscle. Okay, so the calcium is going to bind to that. The calcium, source of the calcium, what was the source of the calcium in the case of skeletal muscle? Yes, please, it is going to be the ECF and the sarcoplasmic reticulum, not the ECF. So I'm going to write sarcoplasmic reticulum only. On the other hand, there are many sources which are going to be present in the smooth muscles. First of all, yes, definitely the ECF calcium will be there. Sarcoplasmic reticulum calcium is going to be there. Okay, sarcoplasmic reticulum calcium It can be because of the IP3DAG, so IP3, right? And there are going to be RYR channels also. And in the smooth muscle, which type of RYR are there? Please let me know in the comments. Which type of RYR? We have discussed that, right? The SM, SM, right? So smooth muscle is there. So what type of RYR are going to be there in the calcium or in the smooth muscles for the calcium entry? Tell me that. Okay, till that time I'll move on forward. The Z line. Z line, please remember, it is a line onto which the actin myosin, they are going to get attached. Okay, and especially in the middle of the eye band, we do have actin. So there is actin will be attached. For the myosin, there will be space. The myosin won't be attached to it. Okay, so space will be there. Then again actin, on the regular intervals, the actin will be attached over there. That is going to be present again in the skeletal muscle, but this organized arrangement is not there in the smooth muscle, so that's why this is going to be absent. Absent. What about tropomyosin? Tropomyosin is present in both. The Papa G is important, right, to control the children's. So Papa G is important. Papa G is present in both, but the Papa G in the skeletal muscle, they are little orthodox, right? So they do not allow the love-in relationship, right? And the tropomyosin in the smooth muscle, they are kind of a modern family type of Papa G, so they allow it. What they are going to do over here, it inhibits the cross bridge formation or cross bridge cycling, right? And over here it facilitates it. It is going to facilitate it. Yes, Kamlesh, you are right. Now, next is the gap junction. Gap junction should not be present in the skeletal muscle. Why? Because understand, I'm writing, right? And I'm going to move my pen accordingly. And as soon as I move my pen, that means the different skeletal muscles are working differently in different force in the different direction. And that I'm going to control because there are no gap junction. If the gap junction is there, what is going to happen? It will lead to what thing? All the muscles will be acting as a single unit, that is the syncytium. The whole, if my hand is contracting like this, one muscle is contracting like this, the whole of the muscle will be contracting like this, which I do not want. Otherwise the fine skills are not possible. What is the use of voluntary control, right? And that's why we do not want any gap junction in between the skeletal muscle. Thankfully we don't have it. Okay, so gap junction are absent in the case of this skeletal muscle. Remember, it may be present or may be absent in the smooth muscles. It may be present, it may be absent. I don't have space over here. I'll write it in the next on the next page. Coming to the junction between the nerve and muscle, what it is called as in the smooth muscle specifically, because I know I'm having that faith that you know what it is called in the skeletal muscle. It is called as neuromuscular junction. But in the smooth muscle, it is not very proper junction, as we do have proper junctional site for the nerve and the muscle where they meet together, right? So that is called as neuromuscular junction, which is not going to be present in the case of smooth muscle. So what is present in the case of smooth muscle? There will be something known as this. I want the answer from you guys. Till that time, I'll discuss the gap junction and then followed by this thing. Okay? Chalo. Let's discuss the gap junction first. In the gap junction, on the basis of gap junction, there are two types of smooth muscles which are present. There are one type which is known as single unit smooth muscle or unitary smooth muscle. There is no division in between them. They are unitary, that means they have united together. That means the gap junctions are going to be present over here. So gap junctions are present, right? And they are going to act as a one single unit, and they are usually present in the viscera. What are the examples of those viscera? Gallbladder, then your intestine, right? Then urinary bladder, uterus, all these things. What about the multi-unit ones? Multi-unit smooth muscles, they are going to have different units. They will be acting completely different units, right? Each of these muscles are going to act separately. There is not going to be gap junctions. So gap junctions are going to be absent. The gap junctions are absent in the case of multi-unit smooth muscles. And what are the examples? The iris, ciliary body, iris, they are the examples of such this thing, the multi-unit smooth muscle, right? Very good, Kamlesh. It is known as neuroeffector junction, or it is also known as the synaptic end passant. Okay, now see over here, what happens is, in the skeletal muscle, you must have seen that there is a little indentation which is going to be present on the skeletal muscle as the axon terminal is going to come near it. Okay, so this is the skeletal muscle wala junction, that is the NMJ. But this is not going to be happening in the case of the smooth muscle. In the smooth muscle, what is going to happen? Ciliary body. Okay, another example for it. Now, in the smooth muscle, there will be muscle like this. So let's draw the spindle-shaped muscle. Okay, and the nerve will be passing by through nearby it, right? As the nerve is passing by, there are going to be a little swollen areas from which the vesicles will be released as the nerve is going to passing by. There is no axon terminal, specific axon terminal ending onto the specific muscle cell. Okay, so these are known as neuroeffector junctions. Okay, or this is also known as synaptic end passant. The fancy word, nothing else, right? So this is known as synaptic end passant, or is also known as neuroeffector junction. Synaptic end passant or neuroeffector junction. Right? Let's move on to the next thing. If the troponin is not present, what it is, how it is going to cause contraction? As you guys told me, that it is going to have MLCK and MLCP. With the help of that, it is going to contract and relax. As you can see over here, that the calcium is going to enter, and what it is going to do, I told you the calcium entry is possible via ECF also and from the sarcoplasmic reticulum also. Now you can see that the calcium channels in the ECF, that means on the sarcolemma, they are opening, but you can see different receptor over here, which is also opening the channels that are going to be the R/R channels, which will be open up in the sarcoplasmic reticulum through the IP3. Okay, directly R/R are going to open, or through the IP3 it is going to open also. Okay, so IP3 is the secondary messenger which is going to be present, which we will talk about these IP3s and other secondary messengers in the endocrine system. So for now, just trust me, there is inositol triphosphate which is going to be present, which will stimulate the R/R, and as soon as they are going to get stimulated, they are going to open up, right? The more calcium entry will be possible, and these are known as the pharmacological effects of this drugs also. Some drugs, they are responsible for this increment in the IP3 DHE, and please remember, because of these drugs, the calcium may be increased in the smooth muscles also. This information we can use for our own benefit to treat the patients, isn't it? So first thing, what is going to happen, as soon as the calcium is going to come into the muscle, the adequate levels are there of the calcium, it is going to bind to the Cal module. I told you that the troponin will be absent. Instead of troponin, we do have right like this. Troponin is not there, so that's why instead of that we are going to have the Cal module. As soon as the calcium is increased, now the calcium calmodulin complex will be formed instead of troponin calcium complex, right? So calcium calmodulin complex will be formed. And as soon as it is formed, what is going to happen? It is going to stimulate something known as myosin light chain kinases. Myosin light chain kinases. Okay. This myosin light chain kinase, what they are going to do? See over here, as soon as these kinases are going to get activated, definitely they will convert this ATP into ADP and PI. And that means as soon as they are going to get converted, all these cocking processes and binding and the pivoting processes, they will be activated, right? So this is very, very important to activate these kinases. And as soon as the kinase is going to get activated, the phosphorylation of the ATP is going to occur, and that's why the ATP will be converted into ADP. And then whenever we want relaxation, we have to activate. This is going to cause the ATP is going to get converted into ADP and PI. So the contraction will take place. The whole story, you know that already. We have discussed this, right? Cocking and what is the role of ATP, ADP, everything. When we want to relax the muscle, at the time of relaxation, what should happen? There is one more thing that is known as MLCP, myosin light chain phosphatase. Aradhna, good evening, bache. Where were you? From 2 days, I haven't seen a message from you. I was missing you. Chalo. So myosin light chain phosphatase. Myosin light chain phosphatase. What it is going to do? Definitely, it is going to attach myosin. It is going to detach the myosin from the actin, right? And so this is going to be happening there, and which is going to lead to the relaxation back again, right? So this is very, very important. Now understand, there is a limited number of ATPs which are going to be present in the case of, there are less number of ATP as compared to the skeletal muscle, which is going to be present in the smooth muscles, right? So less ATP comparatively, as relatively less ATPs, that is relative to skeletal muscle. And we know that ATP I have written over here, which is important for the relaxation, right? Which is important for the relaxation, isn't it? Relaxation, right? Then there is a second thing which is going to happen. Understand, as soon as the potassium start moving outside, that means the repolarization has taken place. Now at the end part, suppose this was the depolarization and this is the repolarization, which is the electrical phenomena followed by the contraction and relaxation, isn't it? As soon as you must have known that this is the depolarization phase, this is the repolarization phase, and at the end part of the repolarization we call it as after D, isn't it? As soon as this will happen, the potassium will start moving slowly outside. The same is going to happen with the MLCP also. Over here, the activity of MLCP is going to be slower as compared to the activity of MLCK, right? So relaxation will be prolonged, right? So as compared to the, I'm just writing in the short over here, that relatively if I talk about the activity during the relaxation, what is going to happen? The MLCK activity is much more as compared to the MLCP activity, which is again important for the relaxation, right? So this is important for the relaxation, right? And if both of these are less in the smooth muscle, it will cause prolonged contraction. But because of the less ATP. That means expenditure of the ATP is also less, right? Prolonged contraction with less energy expenditure. That means in the less salary as the government asks us to work, right? In the same condition, this is going to happen. We are like a smooth muscle of our country, right? We are working like 24 hours, but we are getting a very less salary which is there out in the market, right? So what is going to happen over here? Prolonged contraction in the less energy expenditure, same as the doctors. So please remember, this is going to be called as the latch bridge mechanism. Right? In the COVID, what they have done, they have prolonged our SRship, they have prolonged our post-graduation itself by the 6 months. Understand, 6 months matters a lot when it comes to, you know, the promotions and everything. But yes, we cannot crib, we have to move forward. Right? I always tell myself that courage to continue matters, courage to continue matters, right? One day the government will realize that doctors are very, very important for this country, isn't it? Let's move on to the next thing, that is length tension relationship. Now we have looked forward into the force frequency relationship. Now we are looking at the length tension relationship. Now till now you all know that in between we do have myosin, which is the thick fiber, and at the side of it we are going to have actin molecules. So these blue color molecules you can see over here, these are actin, and in between these molecules are the myosin. So actin and myosin fibers are shown over here. Now two types of question can be asked in this scenario. What are the two types of question? First, they can ask you what is the optimum length, where the active tension is maximum, that is known as optimum length. And you guys already told me what is the active length of or active tension which is going to be maximum at the length, that is going to be 2 to 2.2 micrometers. That is the optimum length, right? And remember that if I ask you, there is going to be a point, they have just mentioned in this graph ABC, but in the exam they are going to give you, let's say this is the A point, this is the B point, this is the C point, and let's say somewhere here it's the D point. They're going to ask you from where to where it is going to be the optimum length. If they are going to give you, in one university exam I have seen this, in the NEET exam also, they have given both the options, that B to C and C to B. Now you get confused. Remember, whenever they ask about the contraction or the stretching of the muscle, the answer is going to change. Okay, how? Remember, on the x-axis they have mentioned the length of the muscle, which is gradually increasing towards the right hand side, right? As you go from the left hand side towards the right hand side, isn't it? And the tension has mentioned on the y-axis. So first of all, x-axis length and y-axis tension, and that's why we are looking after the relationship of both. So that's why it's the length tension relationship, right? Today we are talking a lot about these things, but anyways, okay. So if the length increases, that means I cannot contract the muscle and increase the length together, right? If the length is increasing, that means I'm doing the stretching of the muscle, right? If I'm doing the contraction, what is going to happen to the length of the muscle? It is going to decrease. So whenever you're going from the less length towards the more length, that means over here in this scenario, from the left to right, there is going to be stretching. So if they ask during the stretching, at what point there is going to be maximum or optimum length which is going to be there, that is, the answer is going to be B to C. If they ask the question for the contraction specifically, in the contraction I know that the length is going to decrease, isn't it? And that's why the answer becomes C to B. That is going to be the contraction point or the optimum length point. It is a useless question, but yes, I have seen this question and the options in many books also, MCQ books. So that's why I have mentioned over here, because again, in the personal messages, there are lots of messages why this is the answer, why that is not the answer, right? So this is the reason why it is the answer. Okay, let's move on. How this is happening. So see over here, everything should be in the range, right? The joke should be in the range, the padhai should be in the range, right? There should not be too much stress in the life, right? Enjoy karna bhi zaruri hai, padhai karna bhi zaruri hai, baki sare kaam, you know what I mean by baki sare kaam karna zaruri hai, right? So there should be someone to tell us, our seniors also, that Saturday Sunday ko chutti chahiye, right? But anyways, padhai bhi zaruri hai in that matter. For that, we have to do everything so as to maintain our homeostasis. Same happens with the muscle also, right? So what is going to happen in the muscle? If you stretch a muscle till a point, it's okay. But if you stretch it too much, overstretching of the muscle, what is going to happen? Definitely the active myosin. Suppose these are the myosin heads and these are the active sites of actin. Okay, suppose that. Okay, if you stretch it, okay, there are still some cross bridges which are there. But if I stretch it too much, what is going to happen? The whole of the myosin heads, they will be get away from the active sites of actin, and there will be no cross bridges. So overstretching will decrease the force of contraction. On the other hand, if I start to contract, so suppose this was the first phase, so I start to contract, first of all, one cross bridge formed, then two, then three, then four, then five, like that. Okay, but if I again, that means the overlapping, I contracted it too much, what is going to happen? Both the sides are the actins are present. The actins start to overlap on each other if you contract it too much. So what is going to happen? Again, if the active site of actin is covered by the another active site of actin, that means there is no cross bridge formation. It should be the myosin which should be meeting with the actin, hai na? So homo wala, that is not natural. I hope you understood this. Okay, so actin should not be overlapped with the actin should be overlapped with the myosin, then only the cross bridge formation is going to happen. See over here in this condition, specifically what is happening? The actin is overlapping onto the actin. That means what is going to happen to the cross bridge formation? The cross bridge formation is going to reduce. It is not going to increase, right? So everything should be in the range. That means everything should be in the homeostasis optimum length ke liye, right? So the overstretching is also not good, and contracting it too much is also not good. And that's why as you start to increase the stretching, the active tension is going to decrease. Active tension is because of the contractile force which is generated due to the cross bridge formation in the skeletal muscle. And what about the passive tension? Passive tension which is generated because of the stretching. And that's why they ask you to do the stretching before the exercise. But that should not be too much stretching. Understand that. Okay. So as you start to contract from the stretch state, what is going to happen? See over here, as I start to contract the muscle, the active tension will start to increase. It will increase in a certain time, right? And after that, it will start to reduce because there is an overlapping of actin by the actin molecule itself, right? And that is why it is going to decrease. Same is going to happen if I start to stretch the muscle from the contracted, fully contracted state. What is going to happen? I'm going to go in the reverse direction. I'm going from the contracted state towards the relaxed state and then the stretching state. So what is going to happen? There will be a point which is coming where the active tension is going to be maximum, and then it is going to decrease. And please remember, the Starling law, Frank Starling law, is based on this only, that in the cardiac myocytes also this is going to happen the same. And that's why we write over there that within the physiological limit, if you increase the input, that means if you increase the filling, your contraction, force of contraction will be increased, and hence the ejection will be increased. That means if you increase the filling of the ventricle, which is going to happen with the venous return, that is the input for the heart, and what is the output for the heart, that is going to be your the EDV is going to be the venous return, and EDV is going to be your input, stroke volume, or cardiac output is going to be your output. Okay, so this is the length tension relationship. Just mention over here, because we have talked about optimum length. It is from B to C or C to B, depending on whether we are talking about contraction or relaxation. Okay, C to B or B to C. Let's move on to the next thing, that is the Henman's principle. Can you tell me by the diagram what you are able to get the message? What is the message in the diagram which is given there? Yes, please, guys. Are you alive? Are you awake? Chalo. So tell me, you can see over here, there is only one plate which is attached, right? So I'll just mark it by this. Only one plate is attached. Then what is happening? There are different motor units which are shown. These are the smallest one, medium size and large one. Okay. So depending on what type of muscle fibers are there, how much weight you are putting up onto that rod on that wrestler. So depending on that, the motor units are getting activated. What are the motor units? At least tell me that in the comments. Okay. So that is going to happen that if you attach less weight, the smaller motor unit will be activated. If you add more weight, what is going to happen? The smaller motor units, they cannot produce that much amount of force to overcome that force. Okay. To overcome that force, we require small as well as medium sized motor unit. But if you attach too much weight, what is going to happen? If you attach many weights to overcome this weight, now the small and medium size, they won't be sufficient. So we have to activate the large motor units also. So that means what is going to happen? First of all, if I start to pick up the pen, the smaller force is enough. Then I start to pick up the glass which is filled up with the water, definitely I need more force. If I start to pick up the chair, then I require to activate whole of my motor units, and I am not sure that will I be able to pick up that chair or not, right? So this is known as the Henman's principle. It is very simple. That is also called as recruitment phenomena in the simple words. The name itself telling you recruitment of the motor units that is going to happen over here. That means as soon as you start to work, what is going to happen? Whenever you need to generate the force in the muscles, the smallest unit will be recruited first. Then come the medium size, and then come the large motor unit at the end. Okay, so that is going to happen. This is the recruitment which is going to happen. Okay, and what about the de-recruitment? When the muscles start to relax, first of all, the relaxation will happen in the large motor unit, then the medial, and then the smallest in the reverse manner. Okay, so this is going to take place over here. Very good, Kamlesh. Very, very good. This is known as motor unit. Excellent, right? So smallest, then the medium size, and then the largest, they will be recruited simultaneously, and this is known as recruitment phenomena. This was given by the Henman's, and that's why we call it as Henman's principle also, right? So the same thing is shown in the diagram also, that as you increase the weights, definitely what is going to happen? As you increase the weights, the motor units that are recruited, they are simultaneously increasing in the size also. Okay. So as I asked and I got the answer also, that the motor unit is the motor neuron, that is the single motor neuron, and all the muscle fibers supplied by it, right? So single motor neuron and all the muscle fibers supplied by it is known as motor unit. Okay, so it is known as the motor unit. Now over here you can see, so there is a single motor neuron which is supplied which is supplying the many muscle fibers, the whole of the muscle fiber, and that single motor neuron will be called as motor units. There will be different combination which will be given. I don't know, I'm not sure if the MCQ is there or not, because I asked them to add, but let's see. There will be questions like this, and answers or the options will be given to you guys. What are the options which are going to be given? That first thing, they can give you one motor neuron, single motor neuron. This will be the option. Then they can give the other options like many motor neurons and one single muscle fibers. One single muscle fiber can be supplied by many neurons, but that is not going to come under the motor unit. Understand? Okay, so we have to make the unity in the muscle cells. So muscle plus motor neuron. Okay, so that is going to be motor unit. Okay, and other neurons, many neurons plus many muscle fibers, one single neuron and one single muscle fiber, all these options will be there. You have to omit that. So motor unit is going to be this one, that is one single motor neuron which is supplied by the, or which is supplying all the muscle fiber. They are going to included in this. Now what is going to be happening in the element lesion? Element means if this motor neuron is gone, okay, so suppose this motor neuron is gone, which is the lowermost motor neuron which is supplying the labors. Who are the labors in our body? They are nothing but your muscles. Muscles are the labors in your body which are actually working for you. Alright, so the lowermost, there is hierarchy. As let's say you are MBBS students, above you there will be seniors, then there will be interns, then there will be PGs of that. Let's say you do your internship and you go in a particular department, so there will be postgraduate residents, then there will be JRs, that means, and there will be SRs, then there will be the APs, professors, director, professor, HOD, dean, and so forth, right? So this is the hierarchy which is there, right? And remember that the lowermost to whom you are contacting is your just seniors, right? So who are the lowermost in this hierarchy in the case of this muscle and neurons? So please remember, the lowermost neuron are the ones which are directly supplying these labors, that is nothing but the muscles in our body, right? So if the lesion is there in the lower motor, so element means lower motor neuron lesion. What are the lower motor? So please remember, whomsoever supplies to muscle. If they are defective, what is going to happen? They won't be able to supply to the muscle. Now what is going to happen? Initially there will be no contraction for some time, okay, because the spinal shock, this is a condition in which the spinal cord is not able to give the supplies to the motor neurons, right? And that's why there is no motor neuron, they won't be able to understand what happened suddenly. Okay, so in the first few moments there will be no contraction, but after that what happens is they start to fire a lot. Why? Because understand that there is no inhibition onto them, and that's why they start to fire, and their fires, the firing of the gamma motor neuron, that is going to be causing the spasticity and the rigidity, but if the whole of the muscle is involved. Okay, but if not, suppose there is a muscle fiber, nerve fiber which is cut in the lower motor neuron lesion, or one neuron is cut which is supplying few muscle fibers, not the whole muscle, then what is going to happen? If it is supplying to the fascicle, and if that nerve is cut, what is going to happen? Definitely there will be fasciculations will be seen because of the gamma motor, increased gamma motor neuron activity. And there is one more condition which is known as denervation hypersensitivity. If you cut the nerve, what happens is that this, again, gamma motor neurons are also gone from there. But understand, there is receptors which are going to get overactivated. There is going to be upregulation of these receptors, right? So this is mainly seen in the something known as denervation hypersensitivity. Right, in that there is something known as upregulation of the receptors which is going to happen. The receptor numbers are going to increase. Even the gamma motor neurons are not there, what is going to happen? Definitely the this upregulation will cause whatever neurotransmitters are released by the side branches, that is also catch up by these receptors, and they start to activate without even the stimulus given to them, right? So because of that you can see the twitchings, Okay, and if you can see by the naked eye, that means this has happened in the more number of muscle fibers, that is the fascicle. So the twitching is going to be seen in the single fascicle. So whole one fascicle, you can say group of the muscles are involved, group of the muscle fibers are involved. And if you see, you cannot see by the naked eye, but if you do the EMG, you can investigate that there can be a fibrillation which is present. Only single muscle fiber is involved. How to remember? No need to even remember. Fascicle means we know that this is the fascicle. That means it is a group of motor fiber, this muscle fibers, and fibril matlab one single fiber. Okay, so this is how you can remember that this is going to take place, isn't it? All right, so last but not the least, the types of muscle fiber. Do you know some types of muscle fiber? Tell me, what are the types of muscle fiber? What are the types of muscle fiber? Do you know? There are 3 types of muscle fiber. Type 1, type 2, type 3 are there. But then there are going to be slow oxidative, fast oxidative, glycolytic. That means it is going to contain both the properties. That is, they are the mixed fibers. They do have oxidative property also, glycolytic property also. Remember, oxidative phosphorylation is going to take time, and that's why these fibers are slow. And when you act slow, you can go long distance, right? So if you have to run marathon, you're going to go and activate the slow twitch muscle fibers, and that's why you have seen that the runners, they do not start to run very quickly. Rather, if they want to go long distance, first of all they are going to run slowly, and as soon as they come towards the end, what is going to happen? Definitely they will start running very quickly, right? But that is going to happen when they come to the finishing line, near the finishing line, not in the beginning, because they do not want to exhaust themselves from the glycolytic the stores, okay, glycogen stores. Now there are mixed fibers which are going to have properties of both, that is oxidative also, glycolytic, but not complete. Some glycolytic property, some oxidative property. Then there is going to be the 2B fibers, which is going to be fast glycolytic fibers, right? They are very, very fast. When you want to run only 100-meter sprint, at that time you do not have time. So that's why whatever store you are having as an energy, you're going to apply that, try to win the race, right? So throughout the year, or let's say you are giving the NEET, you're preparing for the NEET, and you do preparing, or you started the preparation in the MBBS itself, you're going to run the marathon. But as soon as the exam comes near you, let's say the exam is in the 1 month or 2 months, definitely you have to start going faster. That means you are going to activate the glycolytic, you have to run the sprint, and who are going to manage both the things, marathon as well as sprint, they are going to win the race. Okay, so the whole thing you have to maintain throughout. Okay, so whenever you are feeling motivated, just study by yourself, or attend the classes, and you do not feel like studying, at least see the videos which are available. Okay, so that will continue your momentum. So this is going to be the 3 types, and in some books only given 2 types, type A and type B. In that they just mention slow oxidative and fast glycolytic. So major differences, I'll make you write down the table also, but just the major differences that we can just rule out by the name itself. Slow oxidative, that means they must be doing oxidative phosphorylation. That means they should have the oxygen, and they are working on the basis of anaerobic metabolism, isn't it? And that's why they are slow. On the other hand, glycolysis, glycolytic fibers, they do not require oxygen, right? They are not working on the oxidative phosphorylation. So that's why they do not require per se much of the oxygen store, and depending on that, their color is going to change. Remember, as in the blood, we are going to have one pigment which is known as hemoglobin, which is going to carry the oxygen with it and which is going to give us the red color to the blood. Okay, and that's why the oxygenated blood is comparatively more red as compared to the deoxy blood, okay, which is usually shown by the purple or the blue color, right? So that's why in the oxidative type of muscle fiber we do have a globin molecule which is present in the muscle. That's why we are going to call it as myoglobin, isn't it? We are going to call it as myoglobin over here, right? Myo means muscle and globin, which is going to carry the oxygen, which is going to combine with the oxygen. What is the difference between the hemoglobin and myoglobin? Please understand, hemoglobin carries 4 molecules. One hemoglobin molecule carries 4 molecules of oxygen. On the other hand, one myoglobin will carry only 1 molecule of oxygen. That's the only difference, okay? There are many differences that it have the oxidative phosphorylation, helps in the oxidative, plus this will release the oxygen at very low PO2 in the muscle, right? So the partial pressure of oxygen, when it is going to go below a certain level, then only this will give up the oxygen, otherwise it won't give up the oxygen very easily, okay? And when we talk about OHDC, that is oxygen hemoglobin dissociation curve, that is going to be the sigmoid shape in the case of hemoglobin. This is going to be parabolic in the case of myoglobin. That we'll discuss in the detail when we are going to go towards the respiratory or blood system. Okay. For now, just remember this myoglobin is present and it is going to bind to the oxygen, and this is going to give a red color, and that's why these are going to be of red color. Okay. For this contraction and relaxation, we require the calcium also, we require the Cerca pump and capillaries. So remember, many other capillaries, easier it is to get the oxygens, right? So the oxygen and the Cerca pumps, they will be activated more and more, right? Which is given in your all the books, especially I have the table from the Genome itself, right? As you come towards the fast glycolytic, we do not require that much of the oxygen, and that's why remember these are going to be white in color. So white fibers are going to be more. So that's why they do a testing, genetic testing and muscle testing, in which they took a biopsy of your muscle, and they will overall analyze. If you go towards the specific scientist, they will analyze what type of sport is better for you. If you do have more of the red fibers, then that means you are able to do a longer time workout more, but it should be slower activity. It is not good for the sprint, it is good for the marathon like types of sports, right? If you have to act very, very quickly, then you should have the glycolytic fibers more and more. So this is the table we are going to make. Just before that, just go through it, which is given over here, that myosin ATPase activity and all the activities, they are going to be slower as compared to the they are going to be slower in the case of slow oxidative as compared to the fast fibers, and that's why in the beginning itself we have written it as slow, right? Glycolytic oxidative, the name itself will tell you. Unit type, they are going to give the resistance also, right? And slow oxidative, fog, and this is FG that we have seen over here. So let's try to write it. Glycolytic on your own, this is the easier format, so I have chosen this format for you guys. So first of all, myosin ATPase calcium pumping capacity, this is going to be slow, as we have seen in this case, slow, high and high. Why? Because it is the slow fiber, that it means it should be slow. Color, we have seen that because of the presence of myoglobin, which is going to be higher in this case. Moderate, because it is going to have mixed fiber, and it is going to be least or lesser, or low we can write in the case of type 2b. Okay, and because of the high myoglobin which is present, it is going to be red in color. This is going to be mostly white, and this is also white. Okay, so the slow, the moderate, and that is going to be white, which is going to be fast, fatigable. Now type is slow, I agree on that. So these are going to be slow oxidative, these are going to be fatigue resistant, and these are going to be fast fatigable. right? I hope you can see that these are going to be fast fatigable. The fatigue will be set in the slow fibers very, very late as compared to the type 2B fibers, where these are going to get fatigue very quickly. And that's why if you do not want to get fatigue early, you have to go slow, right? In the case of running, I'm talking, not in the case of studying, right? Glycolytic, as the name itself suggesting us, glycolytic activity will be higher in this, and oxidative activity is going to be higher in this. Both the activities are going to be moderate in the case of middle one. This is going to be low over here, this is going to be low over here. Easy? Very easy. What are the examples? Remember, you don't run marathon all the time, right? But you maintain your posture all the time. If you're sitting or you're standing for a prolonged time, that means you are maintaining your posture, and anti-gravity muscles such as the back muscle is going to help us in that. So the back muscle, which is going to act as the slow oxidative, or any postural muscle, I can write any postural muscle for that matter, which is going to not have the fatigue very easily. That's why we know that they are going to activate through the oxidative phosphorylation, right? What about the fast oxidative glycolytic, the soleus muscle? You can maintain your sitting posture for long, but not the standing. Why? Because the soleus muscle, they do have moderate type or both mixed type of fibers are there, which are going to get fatigue ultimately. Okay, then comes the fast fatigable, in which muscles you do have fatigue very quickly. The hand muscle, and that's why you ask PDF all the time, right? Why? Because the fatigue will be set in your hands very quickly, and that's why you cannot make the notes. So hand muscles, please remember. Not only that, the extraocular muscle, as soon as the class start, everyone is having their eyes wide open, but as soon as the class come towards end, everybody will be like this, right? So that's why the fatigue is going to set in the hands and extraocular muscle very, very quickly. Okay, so this is the whole thing that I wanted to mention. So I feel this chart is much easier as compared to the other chart, and it contains all the information that is required, because even in the genome that I showed you just now, the chart was given, the mitochondria has not mentioned, capillary has not mentioned, and as I had told you, the cytochrome oxidase, all these things are require more to provide the oxygen to the muscle, and where the oxygen is required, wherever we are doing the aerobic metabolism, and that is going to happen in the slow oxidative format, right? So please remember, so fog FG. Okay, so that is the easiest mnemonic possible, and this is why this is going to take place. Okay, so this chart is much more easier. I feel it is easier. If you can remember the other chart, you can remember, just remember the extra points also. Okay, we haven't mentioned the size. If you want to remember, small, large and large. So in the color itself, you can write red small fibers. These are going to be smaller fibers. These are going to be large fibers, and this is also larger fiber. To activate faster, you need the less resistance. So that's how you can remember large and large. They are going to have, or they are going to get activated very, very quickly. Okay. So that is all. Membrane potential, we know we are talking about skeletal muscle. So what they are talking about is the RMP. We have discussed the RMP is going to be -90 for all. It is not going to change depending on the fibers. If it is skeletal muscle, it will be -90 itself, right? So that is all about the lecture. Just give me the answers quickly of these questions, and then we'll finish up the class. Okay, first year students were studying about the exercise physiology. Oh my god, you guys are very, very interested in the studying, right? So you started to study exercise physiology. Let's see. They came across the concept of recruitment and principle of Henman's, that is Henman's principle related to the motor unit. Okay, so the hint over here is given, that is Henman's principle or recruitment phenomena. Okay, the motor unit consists of, so what they're asking is motor unit consists of. Chalo, give me the answer of first question. Let's say this is the first question. All muscle fiber in the muscle, motor nerve and the muscle fibers that it supplies, afferent neuron center and efferent neuron, single muscle fiber, all neuron that innervate it. Answer, bacho. I'm going to move ahead. Next question, which came in AIMS 2017, I have said, I guess, AIMS 2018. Okay, so this question already I told you the answer for this, but if you remember, 23 years old male present to OPD with a complaint of progressive muscle stiffness, cramps, with a history of continuous twitching of the muscle. On clinical examination, there was atrophy of the muscle with the delayed muscle relaxation. The hint over here is this. With the DTR, okay, that means deep tendon reflexes. What is DTR? DTR is, it's not BTR. Deep tendon reflexes. Yes, I'm getting the answers right. So I got the answer for our first question. What is the answer for the first question? Yes, you are absolutely right, Kamlesh. Where are the other students? Why you guys are not giving the answers? We know that the single motor neuron, we have discussed that. Single motor neuron supplying all the muscle fiber and all the muscle fibers supplied by it. Right, so whatever fibers are supplied, so not only all the muscle fiber, afferent neuron and efferent neuron की हम बात ही नहीं करते हैं, and all neurons, right? This is not possible. What about the second? Yes, I got the answers over here also, right? So what is the answer? Which is this condition? This is nothing but the Isaac syndrome. Yes, potassium channel. I told you the repolarization will be followed by the relaxation, and that means the delayed muscle relaxation is there, and that is means the repolarization is not happening properly, right? Depolarization is not proper or defective, and that means potassium channels are defective. Voltage gated, I'll just write VG. KC, that is voltage gated potassium channel. What was the syndrome? The syndrome was Isaac syndrome. Isaac syndrome, which is going to have the antibodies against the potassium channels. So there are going to be antibodies against the potassium channels. Okay, so the potassium channels will be destroyed, and because of that the repolarization will be defective. Let's see the options. Etiology is due to voltage gated sodium channel. Understand, voltage gated sodium channel nowhere we have mentioned, right? And if the voltage gated sodium channel is defective, what is going to happen? There will be no action potential production, and that means cramp is not at all possible. Twitching of the muscle is not possible. DTR, deep tendon reflex, is not possible. For everything you require a stimulus. If the sodium channels are affected, there will be no stimulus, no contraction. So if no contraction, from where the relaxation will be delayed, right? So this is absolutely false, and what they have asked us, accept. So even if you do not know, you can just rule out via whatever information you are having. Smooth and cardiac muscle are usually spared. I told you, thankfully we don't have that in the smooth muscle or the cardiac muscle. Otherwise we will go in the cardiac arrest, right? If the delayed relaxation is there, that means filling won't be happening. The muscle will be continuously contracted, and that will lead to the patient going into the cardiac arrest. And thankfully we don't have such thing in the cardiac and skeletal muscle. That is true. It affects the peripheral nerve. That is also true. Central nervous system is usually spared. And yes, one more point you can note it down, that paraneoplastic syndrome, that means it is going to cause malignancies. That means tumors and cancers. It is going to be associated with the various tumors and cancer can be there. Okay, may or may not be there. So you can add this point where we have learned about the Isaac syndrome. Okay, so even if you do not know the syndrome and everything, how to rule out, you know that. Okay, so I guess that is all for the class. I'll just go before going, because this is the first Prof session. So with the MCQs, I'm just wanting to explain more about the questions that are going to come in the exams. So you are going to be asked about each of the proteins. So please remember actin, myosin, troponin, tropomyosin, desmin, alpha-actinin, capsi protein, and the titin is also known as connectin. What is connectin? They can ask you about the connectin. The connectin is the another name for the titin. Right, you have many names. Your friends call you by some name, right? Your girlfriend, boyfriend, whatever, they are going to call you by some name. Your mom and dad calling you by some name. On the Aadhaar card you have something other name, right? So all these names. So similarly the titin is having a nickname, that is connectin. Why? Because it connects the Z line to M line. Okay, so they can ask you about that also. So all the protein we have discussed in the brief, right? So this is going to be a short question, not a long question. Very long question won't be there. Then that will be describe the mechanism of excitation contraction coupling. Either they can ask you in the skeletal muscle, excitation contraction, and the cardiac muscle, or what they can do is they can ask you combinedly both or the differences between these two. It can come as the long question also, so you have to prepare the notes. If that comes, you have to draw the diagram. Please remember, if the question is coming for 4 marks, at least draw some short diagram, chota sa diagram, any diagram is sufficient. If the questions are coming from for like 6 marks, 8 marks or 10 marks, please draw at least 1 or 2 diagrams with a neat labeling. Labeling is important. If you just draw circles and triangles, that is not going to give you a mark, right? So definitely you have to write it with the all properly labeled diagrams should be there. Okay, if the labeling is not there, no examiner is going to give you the marks. Okay, because the copying is easy, but we know that this writing part is going to be difficult, right? The circles and the waves are not going to get the marks. Next question, schematic diagram of ATP and cross bridges. You just can draw like this, the myosin head and neck and then the body, right? And then you can show myosin ATP is over here, and via the bands you can just draw the actin fibers over there also, right? Okay, so this is going to be there. So you can just draw the ATPs are attached or ADPPI are attached, what is attached there, right? And you can just make a circle of it. We have discussed that if in the simplified format ATP is attached, it is going to be detached from the actin molecule if new ATP is attached, right? If you are going to say that the ADP and PI are going to attach, what is going to happen? There will be cocking, but calcium is attached, then it is going to, calcium is there, then it is going to attach to the actin, and as soon as they both are going to get removed, it is going to pivot. The neck is going to pivot, and which is going to drag. If it is attached with the actin, it is going to drag the actin centrally. Okay, new ATP will come, and that is again going to detach these molecules. So this is going to be the ATP role you have to write. Next question, describe the organization of the muscle fiber and fibrils in that. So first of all, you can start with the myofibrils that we have discussed about, the fibrils and their coverings, and then fascicles, then group of the fascicle, that is the whole of the muscle, right? Then you can do about the muscle fibers. If they have asked about the fibers, actin, myosin, everything you have to write. You can mention the parallel and series elastic elements over there, components over there, if it is for the more marks. Okay, write a short note on contractile protein. You know contractile proteins, there are nothing but the 2 contractile protein, actin and myosin. You can write anything about them and everything about them, okay, depending on the marks, right? And give their functions. If it is for more marks, definitely you can write the regulatory proteins also with that. Label diagram of sarcotubular system and sarcomere. Sarcomere, that means you have to write about actin, myosin and everything, right? Z line, M line, you have to show all the zones, all the bands, all the lines, everything, right? Then comes the sarcotubular system in the muscle. You have to mention whether it is striate, diad, what it is. And remember, in the smooth muscle, I told you that the Z lines won't be there, and instead of Z line, what we have is the dense bodies. What are the dense bodies? Suppose this is the spindle shaped smooth muscle. What is going to happen is that from one side there are going to be many actin fibers which are going to come, and they are going to be coming or originating from one side. So that's why that looks very dense, and this is known as dense bodies. So instead of Z line, we do have dense bodies in the smooth muscle, right? And in between there are going to be myosin fibers which are going to be present. So there is no proper alignment, there is no proper arrangement of the Z line and the M lines specifically in the smooth muscle. So you have to mention accordingly what they have asked you. Okay, so this is the difference between sarcomere of the smooth muscle and the skeletal muscle. Role of the sarcotubular system in the contraction and relaxation, we have discussed that in the case of the skeletal muscle versus the cardiac muscle, in which cardiac muscle we require the N6 also. Okay, then the mechanism of calcium release and uptake of this sarcotubular system, we have discussed that also, how the calcium comes and goes away, right? And if it is for 8 to 10 marks, please write applied aspect. You cannot forget to write the applied aspect, otherwise the marks will be cut, because they have specially mentioned that it is the integrated learning that we have started. And I would like to take a leave of yours with this wonderful message, that is, sometimes the smallest step in the right direction ends up being the biggest step of your life. So add up the knowledge little by little, and that is going to end up giving you the good results, right? So that is all from my side. I hope you like the session. Let me know in the comments, right? Thank you, and thank you. Bye bye, take care, good night.